Very good morning, everyone. Welcome you all to the second International Conference on Clinical Nutrition and Dietary Lifestyle, organized by Universal Society of Food and Nutrition and associated with Biotics. I'm sure you had a great day yesterday with us and hope to see the same enthusiasm from all the presenters and attendees today. I would like to welcome all our honorable dignitaries and eminent keynote speakers. Bijoy Bhattacharjee, founder and consultant dietitian, RB Diet Clinic, assistant professor, MIT University, Kolkata. Our next keynote speaker, Mr. Eminch Kaul, Director Food Safety and Sanitation, APMEA, Kerry, Malaysia. Our last keynote speaker, Mr. Jason Freeman, founder and CEO, Farmer Direct Organic, Canada. And our session chairs, Dr. Archana Annapurne, Director, School of Beauty, Wellness and Nutritional Symbiosis Skills and Professional University, India. Next session chair, Garima Upadhyay, Professor Home Science, Food and Nutrition, Consistent College of Banaras Hindu University, Uttar Pradesh, India. Our last session chair, Dr. Poonam Jatwani, Assistant Professor, Allied Health Sciences, Jaipur National University, India. I welcome you all once again to the second International Conference on Clinical Nutrition and Dietary Lifestyle. I hope all will have a great day with us today. Now, I would like to welcome our keynote speaker of today, Bijoy Bhattacharjee, founder and consultant dietitian, RB Diet Clinic, assistant professor, MIT University, Kolkata, India. Ma'am, over to you, please. Ma'am, you're on the mute. Just give us a moment, ma'am.
Thank you, Joshua and Biolix, for giving me this opportunity to be here today amongst everybody and to share my views on my topic. I believe I am audible.
Yes, ma'am, you are audible.
Okay. Thank you. Thank you so much for your kind introduction. So, as we would be talking on the various aspects of the clinical nutrition and its implication on the various of the lifestyle. So, the topic for my lecture is going to be the geriatrics, which is a very strong population, but mostly uncovered under any insurance. So, probably the question could have been asked like, why I have chosen the geriatric or the aging as the topic, because it is always better to go for a healthy aging, which can actually minimize the health cost. So without much wasting further, so I'll be sharing my screen. Is my screen visible? Okay. So basically, I'd be talking on the gut microbiota and aging. The gut microbiota is something that we all are aware. I mean, whoever is present here, I'm sure that all of us are aware about the gut microbiota. And recently, we are talking so much about the gut microbiota and its application in various of the things. So I'll be talking and throwing some light on the aging perspective of the gut microbiota. So if you really see that more than 70 million 16S RNA gene sequences, like which have already been characterized in the human microbial composition, are basically residing in 15 of the anatomical sites of our human body. And these microbiota, they play a very immense role in the development of the various diseases, in the treatment of the various diseases, and it's starting from the metabolic syndrome, obesity, cancer, and whatnot. And as we know that a child which is born through a normal delivery get in touch with the various of the microbes or the microbiome, or they get enriched with the microbiome if they are being born through the vaginal delivery. So if we really see that what types of microbiome, in nutshell, I'd just be mentioning, like in mouth, we have the oral microbiome, then we have skin microbiome, we have urogenital microbiome, then we have digestive tract microbiome. Generally, all of us, we talk about the gut microbiome, because that is something that has got the strongest implications. But having said this, the microbiome which are residing in the other anatomical parts, even they are also releasing their metabolites. So as even they are also releasing their metabolites, so they are also regulating our body system. So aging in very nutshell, we know like it's a progressive loss, and it's of the homeostasis, impaired function, and vulnerability to death. So aging, it starts from conception and it ends at death. And the requirement of the nutrition, the requirement of the diversity of the microbes, it also changes with the as a person age. So there are various age-related diseases that include various of the neoplasms, infectious diseases, metabolic, degenerative disorders, along with the frailty, that is the loss of the balance, coordination, muscle mass, and also there is a cognitive decline. So at a fundamental level, the molecular and cellular hallmarks of aging in mammalians have been identified, but these are accompanied by the changes that are associated with the microbiome that we are already having in our body. And this microbiome some way is also related to the various age-related disorders. So how they are getting related, so I'll be discussing one by one in the next slides. So this aging and gut microbiome, there is an interlink. So basically the gut microbiome, we know it is associated with various of the features, like it maintains the gut barrier integrity. Then it also maintains the anti-inflammatory status. Even sometimes it is responsible for the inflammatory status of the body also. It also takes care of our immunity and cardiometabolic health, and also the major part, it is taking care of the gut-brain axis. But these old age related clinical issues, then polypharmacy effect, wherein with the aging a person is subjected to various of the medications, is actually leading to the decrease in the diversity of the microbes. And with the more the decrease in the diversity of the microbes, the more the Pandora's box gets opened, because that is how a person's suffering gets more aggravated. So with the aging, if we really see the senescence of aging, so with the aging, if it is unhealthy aging, then it can lead to gut dysbiosis, because the infections would be more, the immune cells are also dying. So the infections would be more, the person would be giving more of the antibiotics, there would be more of the bile disorders, digestive systems are at risk, colorectal cancers can happen, cardiovascular disorders are taking place because of the unhealthy lifestyle, Parkinson's, Alzheimer's, all these things, we know these are age associated diseases. Then we have the hypertension, dementia, all these are also being associated with the gut dysbiosis. So it's a bidirectional association that has been found with the gut dysbiosis and with the aging. So gut dysbiosis is also triggering these things, and the aging is also triggering the gut dysbiosis. Similarly, if there is gut dysbiosis, then it is going to reduce the microbiota diversity, because one particular species is going to be very dominant, right? Or maybe two or three very species is going to get dominant. Then there are the gut pathobionts, like which are going to create the diseases. Then the gut permeability increases, gut motility gets decreased. So constipation is one common problem with the old age people. Metabolism gets decreased. There is a lot of inflammation. Immunity gets decreased, and medication and drugs dependency increases. So all these things is also contributing to aging. So it's a vicious cycle, as you can see in the diagram, is actually going on. So the development of the gut microbiota with the age, how it happens. So there are two varieties. Basically, one is alpha diversity and another one is your beta diversity. So alpha diversity include the species richness within a single host or in the microbial environment. But the beta diversity is the diversity in the microbial community, means the different types of microbial community which are getting enhanced in the system. So these gut microbes that we have, they are not aging, but the incidences of the comorbidities, diseases that are associated with the gut microbiota, they tend to increase as a person or as the host individual is growing older. So there comes a terminology that we really need to focus on, is the immunosenescence. And this immunosenescence, it increases with the age. Basically, it's a process of dysfunction in the immunity that is occurring with the age, and it also includes the remodeling of the lymphoid organs, or changes in the, due to which there is a changes in the immune function of the elderly, and also it is closely associated with the development of the various autoimmune diseases. We know anyways that increases a lot of inflammation, malignant tumors, and also development of the infections. So if you see this is a graph and like how the microbiome composition, it changes with the ages. So if we really see the like in the gestation period, in the gestational period, the microbiome composition is going to get regulated by the type of maternal diet, then how much weight during the pregnancy a person is gaining, whether at all the pregnant women is taking some certain medications or not, or the bacteria, what type of bacteria is there in the pneumatic cavity and in the placenta, and what has been the lifestyle and the hygiene practices that the mother has been following. So if you see, the alpha diversity of the microbiota is less here, means the species specific growth of the microbes is less here. Talking about the parturition, when the mother has delivered, so during that time, what is the gestational age, like is a preterm delivery, or what is the mode of delivery, because the vaginal delivery is going to increase the microbial diversity in the child. However, if it's a cesarean section, the C-section, then in that case, the child is not getting exposed to so much of microbes. So you can see the alpha diversity is at a very maximum peak at once when it is at infancy to puberty, because therein the child is subjected to various of the things. The child is having breast milk, the fatty acid composition of the milk is also deciding what shall be the microbiome content. Then the infant formula that the child is having, the weaning, there are pets, if the child is growing with the pets, if the child is there in the dust hygiene, or what type of probiotics the child is getting exposed to. So all these things basically increases the bifidobacteria count, especially during the infancy. At the puberty, there are various dietary factors, because now the child has started eating various of the other outside foods also apart from the household food. Then there would be, the child is also dealing with the environmental change. So there would be illnesses, fever. So they would be subjected to antibiotics, drugs, antibiotics. Then there can be malnutrition. All these things are going to regulate again the microbiome composition, and it changes. So here the Firmicutes bacteroids are at a very peak. Now at the adulthood, the diversity is the most. If you can see the graphs, the diversity is very high at the adulthood. But with the aging, the immunosenescence is high, the biodiversity, the beta diversity is decreasing because of the lifestyle habit, type of diseases, medication the person is having. And if she is female, then with the menopause, there would be gain in the body weight. With the gain in the body weight, again, the obesity is associated with the gut dysbiosis. So all these things needs to be taken into consideration, like how the diversity of the microbiota, it changes with the age. So this is one healthy intestine, and this is one senescent intestine. So basically, we all know what it is, but still I'd just like to say in very briefly, in healthy intestine, there is a homeostasis. As you can see in the diagram, the columnar cells, they're so tightly joined together, right? So they are tightly joined together, intestinal epithelial cells. And there in the outside of the cells, as in the lumen, you can see like there the various intestinal commensal or the pathogenic microbes are there, right? But when there is the aging process is starting, and if there is gut dysbiosis, hormonal dysbiosis, with the immunity decline, with the stress, because so much of stress of the life is there, with illnesses, organ malfunction, then muscle loss is there. This muscle loss itself give rise to the inflammation. All these things is changing the diversity, and due to the change in the diversity, the intestinal barrier integrity gets lost. With that, we know that the pathogenic organisms that enter, so we call it as a leaky gut syndrome, which is affecting the gut-brain axis, blood-brain barrier, leading to depression, more of hypertension, anxiety, various of the other health issues are being taking place. And also there is a risk of the autoimmune disorders, allergies increases, and all the inflammatory disorders. And most of the disorders are associated with inflammation. So all these things, they start aggravating with the gut dysbiosis. So this aging gut microbiota and the effect on the nervous system, if we see, then we know like how gut-brain axis works, how our gut is regulating our brain and how our brain is regulating the gut. So this dysbiosis partly underlie the progression and the pathogenesis of the various of the metabolic diseases that are actually prevalent in the aging population, like adiposity. This adiposity has been found, like recently a paper was published on it also, that in the Aspen journal. So this adiposity has been found to be associated with the release of the pro-inflammatory cytokines from the adipocytes. Now with the mode of the adiposity, because with the aging population, what does they do? They do not feel like to go outside. So more they are becoming inactive, more there would be the loss of muscle. Why? Because more there would be the obesity, weight gain, because of visceral adiposity would be higher. With the visceral adiposity, there would be release of the pro-inflammatory and inflammatory cytokines. These cytokines are responsible for the rise in the inflammation, leading to the breakdown of the short muscle fibers, leading to the frailty, leading to the sarcopenia. So this is another way how the aging population is getting succumbed to the frailty, and how this inflammation is actually leading to the gut dysbiosis. So probably if we can increase the probiotic content in the diet that I'm going to discuss in the later slides, so then in that case, the patient's diet can be, or the old day or the aging can be controlled a lot more. So this dysbiosis in the gut microbiome and the microbial metabolites that are getting released are actually being known to be associated with the aberrations of the gut barrier integrity, and they also enhance the pro-inflammatory cytokines. So this is another thing. And also as there are alterations in the aging, so the gut microbiome does impact the gut brain axis, so thereby it is hampering the neural, endocrine, nutrient and immunological signals between the gut and brain via enteric nervous system, and can play a role in the development of the diseases of the CNS, like multiple sclerosis, autism, depression, and anxiety. So this is the same thing that you can see, like how it is affecting, that I have already explained. Now, with the microbiome and the host, the major connection is in between the microbial metabolites and the microbiome host signaling. So as we can see with the, as we are aging, so this pink part is the unhealthy aging. So the unhealthy aging is highest if there are pathobiomes. And if this unhealthy aging will be decreased if we can increase the count of the protective taxa, means that if we can increase the count of the protective microbiome. The inflammation, if it is high, then it can lead to the aging. And also if the microbiome, if it is not in, or if there is gut dysbiosis, then it can also lead to frailty and degeneration. So chronological age is accompanied by changes in the host microorganism homeostasis that determines the physical and cognitive decline. So lifestyle and the environmental factors that affect the microbiota, and these factors can delay healthy aging or accelerate unhealthy aging. So basically this in turn deteriorates the life expectancy of the host. So this gut microbiome and the changes which are associated with aging. So there are a few studies that were conducted. So one microbiome studies, they focused on the older people who could be classified into two broad categories. One was like who were reporting the differences in the gut microbiome composition, like with age, what is the gut microbiome composition that was getting changed. And another was a group, they were being checked for the alterations in the microbiome which were associated with the aging linked disorders. Then there were again primarily two categories of the gut microbiome versus aging studies that were conducted in humans. So who studied that what are the gut microbiome composition across the age landscape, and what are the alterations that have been seen apparently between the healthy and unhealthy older individuals due to the presence of the microbes. So they investigated. And based on this extensive literature survey, again, there were two kinds of studies which were seen. Like one group they studied, there were three major groups of taxa, which were showing consistent patterns of alteration, and they were increasing or decreasing abundance with the unhealthy aging, which could be identified. So group 1 taxa with a decreased with the age and were associated with healthy aging. Group 2 consisted of the pathobiomes, means which were causing the disease, that increased with age and were associated with unhealthy aging. And the group 3 population were increased with age and were observed to be depleted in unhealthy aging. So this was the study that was published in the Gastroenterology and Hepatology in 2022. And again, it was done by Ghosh et al. So very recent study. So as you can see, the variability of the microbes, the changes with the age, and the firmicutes are there in the elderly also, in adulthood also, in childhood also, and in infancy also, and the bacteria also. So as we are aging, this variability in the microbes, they're also changing, which are somewhere responsible for our, like, how we are going to respond with the various of the disorders. So there were again few studies which were conducted, like how the dietary habits on the gut microbiota are getting influenced. So we talk about the traditional microbiota, then the low fat, low sugar, high fiber, low added salt, and the low ratio of N3 and 6 were actually found to be improving the traditional microbiota. Whereas the talking about the industrialized microbiota means the microbiome which got altered due to high fat, high sugar, low fiber, high added salt, and high ratio of N3 and 6 profile. So regular exercise and physical activity was something that was found to be giving the better life, quality of life, and it was positively impacting the gut microbiota. Also was also improving the short chain fatty acids and was boosting the intestinal mucosal immunity. This frailty, as we talk about, because it increases the risk of the adverse outcome, like which include falls, then delirium, disability, and older frail adults has been found to have a lower gut microbial diversity than the less frail adults. So another thing is the study was done, like the patients who were treated with antibiotic treated group, they were having the highest abundance of the bifidobacteria in the community dwelling group, and it was lowest in those in the long-term residential care. But with the group of the patients who were not treated with the antibiotics, for them the lactobacilli were higher in the rehabilitation, and with that the hospital stay and the hospital duration was less than 6 weeks as compared to the long stay of the community dwellers, which itself says if at all we can take care of the gut microbiome, then we can probably bring down the hospital expenses or the medical expenses which are associated with the aging. So there are various evidence-based studies which are showing that the genera that are generally found to be higher in the oldest group rather than the younger group are like, these are the species. I'm not reading out the names, as you can already see from the slides. And the genera that are found to be lower in the oldest old people than younger age group are this. So as now we know, like, which microbes are good and which microbes are bad. So probably if we can actually supplement the diet of the aging population with those kind of healthy microbes, then probably we can bring about certain changes into their gut microbiome, and the aging can be delayed. So this gut dysbiosis, it is associated with aging. So in nutshell, if we have to see, so if there is a age-related gut dysbiosis leading to the intestinal barrier integrity gets declined, which is leading to the translocation of the microbes and the microbial metaparticles and metabolites through the intestinal epithelial lining, leading to leaky gut, leading to increased antigenic load, and the systemic immune activation is taking place. As a result, the microbial diversity gets further reduced, leading to overgrowth of some of the distinct microbes and the metabolism instability, which can further lead to the chronic inflammation. And in case of chronic inflammation, again, there would be the continuous breakdown of the muscle fibers, leading to the frailty. So that is how inflammation is also associated with the muscle wasting, sarcopenia, and we know, like, sarcopenic obesity is very critical in the aging population. So this gut microbiota, like the short chain fatty acids, are actually creating the barrier. So they are the major substances, like the butyrates, the propionates. They are the major substances which are taken up by the colonocytes of the colon, and which supplies the colons with the energy, and thus it strengthens the gut barrier. So these things needs to be focused on. So healthy aging versus unhealthy aging, if we see, so healthy aging is someone having someone with the better longevity, good physical activity, for the person who is having good diet, with the microbial diversity is there, and the low cognitive decline. But the unhealthy aging is associated with higher amount of pathogens, cognitive disorders are there, cardiovascular diseases are at rise, CKD, obesity, low physical activity, medications, osteoporosis, and whatnot. So inflammaging is the key that we need to consider while considering the diet of the aging population. So this inflammaging means aging plus inflammation. So it's a low-grade pro-inflammatory state, which is basically characterized by the aging process that include the macrophage activation, cellular stress leading to the oxidative stress, and the genetic factors. So due to all these things, there is an elevation in the circulating pro-inflammatory cytokines that negatively affects the metabolism, decreases the bone density, exercise tolerance, strength is getting released, muscle loss is also there. So this inflammaging is responsible for Alzheimer's, visceral obesity, cancer, diabetes, multiple sclerosis, heart disease and osteoporosis, and vice versa. So this gut microbiota modification can actually alter the inflammaging, which can finally alter the neurotransmission, because with the inflammaging, the gut-brain axis dysfunction also takes place. So lack of Akkermansia study found that lack of Akkermansia and the increased abundance of TM7 bacteria and proteobacteria was actually associated with the activation of local and systemic inflammatory response. So this gut microbiota structure, they basically promoted the small intestinal inflammation, and the T cells got activated in the systemic compartment. And inflammaging is basically induced by the lipopolysaccharides which are present in the bacterial wall, in the gram-negative bacterial wall. So the alteration in the gut microbiota, you can understand that this can actually bring down the inflammaging. So the individual age-related changes that are being seen in the gut microbiome is basically this gut microbiome gets altered with the smell and taste, dentition. People are not eating properly. The digestive enzymes are getting decreased with the aging process. Mucosal immunity is getting delayed. Then your colonic motility is reduced. Then there are various age-related disorders also that are associated with the microbiomes. So all these things can be considered or can be taken into consideration by proper administration of the personalized intervention, like which include the inclusion of the probiotics, prebiotics, especially for the population, for the females who are at the perimenopausal age, and for the male, for everybody rather. Like the moment they have crossed 40, so they should be, they should ensure that their diet is having probiotics, prebiotics, omega-3 fatty acids, symbiotics, psychobionts, and antioxidants. And exercise is something that we have seen that is actually increasing the gut diversity, the gut microbiota diversity. And Mediterranean diet we know that various of the studies have already given their thumbs up for the Mediterranean diet, which has been found to be increasing the gut microbiome composition. So fibers, yes, they increase the bifidobacterium, increase the lactobacillus, and reduces the pathogens, and decreases the inflammation, decreases hypertension, decreases obesity. So the diets which are rich in fiber, like beta-glucan, arabinoxylan, galactomannan, and pectin supplements, are basically the substrate for the health-promoting bacteria, the bifidobacterium and lactobacillus. And saturated fats, we know, increase the firmicutes, increase the proteobacteria, reduces the bacteroids. As a result, obesity is on high, hypertension is there, atherosclerosis is high. So we also need to know that these fibers and saturated fats, why they're important, not just from the disease perspective, but from the microbiome perspective. Then polyphenols should be there in the diet of the aging population, because that is going to increase the bifidobacterium, reduce the firmicutes, and reduce the clostridium, which are the pathogenic microbes. So this, as a result, is going to reduce the inflammation, and the body's antioxidant status would be increased. So there are a few examples, as you can see, like the L-arginine, tannins, and polyphenols that are present in the walnut, pomegranate, raspberries, which are modified like by the microbiota in the intestine, and they are better, they do a better activity in the body also. Then, as you can see, the urolithin, they basically inhibit the formation of the pro-inflammatory compounds. Then resveratrol and curcumin, they exert the changes in the intestinal microbiome. And then quercetin, caffeic acid, rutin, they also influence the health by proliferating bifidobacterium and by diminishing the firmicutes, bacteroidetes, and by stimulating the production of the short-chain fatty acids. So this is another reason why the addition of this polyphenol should be there in the diet of the aging population. Omega-3 fatty acids, we all know that it is anti-inflammatory, but it also does increase the levels of LPS suppressing bacteria like bifidobacteria. So omega-3 fatty acids are not just anti-inflammatory or reduces the cholesterol, but they also do increase the bifidobacteria, thus decreases the levels of LPS producing bacteria. Then symbiotics, they are the mixture which are comprising the live microbes and substrates that are selectively utilized by the host microorganisms, and they do confer various of the health benefits to the host. So they manifest their action throughout several of the physiological and sometimes pathological processes also. So they do the tissue remodeling, Then cancer, injury, aging, and function as a cellular defense mechanism are they also to take care, and they prevent the cellular damage also. Psychobiotics are the recent terminologies that are moving in the market a lot, because psychobiotics are those live organisms that when ingested in good quantity produces certain health benefits for the patients who are suffering from some kind of psychiatric illness. And we know with the aging population, there is a matter of loneliness. They feel lonely, the depression sets in. So that is one reason why psychobiotics more should be introduced into the diet. Also, the psychobiotics, they produce and deliver neuroactive substances like gamma aminobutyric acid and serotonin, which acts on the gut brain axis. So the onset of depression and neurodegenerative diseases are also linked with altered metabolic conditions, and is frequently in the elderly, which we have already seen in the previous slides. So the psychobiotic supplementation, if you talk about, then it should contain the lactis acidophilus, L-casein, bifidobacterium for at least 12 weeks to see some positive effects on the cognitive function. It has also been proven to be beneficial for the Alzheimer's patients. We have curd at our home. That curd itself contains the bifidobacterium and lactobacillus. So by adding the curd every day in the meal of the aging population can actually improve the health, improve the mind of the population also. So overall, if the gut is healthy, so healthy gut is equal to healthy aging. Thank you.
Thank you, ma'am. If any of the participants are having questions, please kindly mention them in the chat box. Ma'am would like to answer all those. Thank you, ma'am. We will move to our next keynote talk then.
Thank you. Thank you so much.
Next, now I would like to call upon our keynote speaker, Mr. Yamnesh Kaur, Director Food Safety and Sanitation, APMEA, Kerry, Malaysia. Sir, over to you, please.
Hello, everyone. Can you hear me?
Yes, sir, you're audible.
Very good. And I'm going to share my screen. Just tell me that you can see my screen. Can you see my screen also?
Yes, sir, it's visible.
I'm going to do sure. So all set to go, right?
Yes, sir.
All right. Thank you. And first, thank you for the team to inviting me for this discussion, and specifically Ramya, who has been following with me from a couple of weeks. I'm sometime delay in responding to the request, but finally, Ramya, I could do this presentation last night. So I'm going to take you through. So what I'm going to talk today is quick introduction. So my name is Yamnesh Kaur, and I'm responsible for food safety and sanitation for Kerry and APMEA region. It's a region that starts from Turkey to South Africa. It covers Nairobi to Middle East, India, Southeast Asia, China, and ANZ. So it's like the whole time zone spread almost 21 hours. Anyhow, so I'll talk you through today few things that Kerry does. I have to talk to Kerry because this is the Kerry time I'm spending, and there's some information also that I want to share with you. And after listening to Professor Bijoya, it looks like Kerry is really helping the community and the people around to be healthy, to stay healthy from when they're born until when they're old and difficult to walk and run. So if you look at it, it's creating a world of sustainable nutrition, and it means something. So we help nourish people, societies. We make sure that our planets are safe. Our planet Earth is safe, and we were making sure that whatever we produce, it tastes well, it is nutritious, and we are not damaging our environment, people, planet, that we have in our sustainability solution that we provide to our customers. So this is what principle that guide us in Kerry. Is inspiring food, nourishing life, and inspiring food is all about innovation, and nourishing is about sustainability. So we make sure that we innovate product that is it's very difficult to have a combination of tasting food and it's also nutritious. So I think at Kerry, we are scientists doing great job by making sure that the both are balanced and people are happy to eat and stay healthy. And nourishing will make sure that we do it the right way, sustainability, less waste that we produce, and we make sure that we don't damage our planet. So the way we produce, consume, dispose of foods is unsustainable for our planet and in people that we all know that, the reducing waste is very important. And what Kerry is doing beyond is we're working on preservatives, so we make sure that our food stay longer. And if it's natural preservative, so it's no impact on the health of people. So we are reducing the waste for different means. That's one of the means by preserving it for longer. So this is how the Kerry looks like when we work on different technologies. So we have done, as a Kerry, we have done 1.2 billion consumer. We are reaching 1.2 billion consumer. The target is to reach 2 billion consumers every day by 2030. We have 147 manufacturing locations, 70 plus technology innovation centers. We have more than 23,000 employees, and we have many R&D scientists that's been working every day with the changing needs of the customer and our consumers. So we all over the world, if you look at here, let me use my pointer. So we all over the world, and we have innovation centers, and we're still growing. But at the same time, we also make sure that we enable our customer to move along the sustainable nutrition spectrum. What that means is that we try to understand what the customer needs and then tie up with the sustainability agenda of this world and how we want to make sure that there's more sustainable nutrition that's created. We make sure that we understand customer. We have food safety and security of nutrition that we produce. We make sure that our formulation is as clean as we can do. And this positive and balancing nutrition that I've talked to you, saying that what population, what zone, what part of the world required what, so it's very balanced and positive. We have proactive nutrition, we're going to talk a little more today, and then we also work on sustainable nutrition. So that's how we create sustainable nutrition, understanding customer need and then delivering them what exactly they need. So we have Kerry Health and Nutrition Institute. We have 150 experts that's been working, and we are also helping industry by having white papers. If you say 22 white papers being released, and there's many articles that keep on going. This is a little old data, maybe a year, 2 year back, but there's a lot of work that our KHNI teams doing day in and day out. And what they do actually is, if you, how to move this slide up there. Let me close this one. All right, I don't know how to close it. Anyhow, so proactive health nutrition. So we work in different streams. And this is something that, we have different brands that in the market. Our BC30 is being used in a different formulation by our customers. We are very serious about what we do about proactive health. And we also aware that when the child born, he has to get into some nutritional prescriptions, and we are very mindful how we create those. So we have few here for the women and infant health. This is LC40. Poly3 is Professor Vijay talked about omega-3. So this is the omega-3. And then we have digestive health. We talk. So it's a very good combination that after the previous session is very much resonating with how Kerry is working towards probiotic, BC30 is probiotic, and how we make sure that the all ages has healthy digestive system. We work majorly on the cognitive health. Again, omega-3, is for a healthy brain. Sensorial is many of you I see from India understand ashwagandha. So this is the extract, the sensorial. You can go on the Kerry side and look at this combination, how it's been produced and what the component. And we also does a lot of work on immune and joint support. There is Valmune. I'm going to talk a little bit about few nutrition here, and then we'll talk about how Kerry produced that in a very safe way for food safety and people safety and for our consumer safety. So there is a few brand here. That's all produced and backed up with a lot of science, food science behind it, nutritional science behind it. And this, as you know, it's a combination of what we're talking to, the combination of pharma and food, right? So this is all those aspects are being contributed and taken care when we produce any of the products. So few like Wellmune, if you know that is proprietary baker's yeast based, and it makes sure that it's used by all the ages to stay well and stay healthy. BC30 is actually probiotic. It's very highly stable spore forming probiotic, and it also the beauty of this molecule is that stay intact and without losing the potency in very harsh manufacturing processes. So that also means that it can be blended and used in a different way in the manufacturing processes. And there are a few of here, but I just want to make sure that I feel why I'm so proud about what Kerry does. We actually every year touch more than 2.2 million babies who's nourished with infant formula. And there are all our infant formulas are producing using our proteins. There are different ways that drinks we use in Kerry that Costa Coffee is using Prodyne. They fortify their vegan-friendly drinks range. And there are many other customers they use our proteins with proactive nutrition in their blends. We have few, we also look at, how we quote our medicine. So this is also some Kerry works to produce product that's more sustainable. And Wellmune, I think I've already talked about this one, right? And there are many manufacturers, industries across the globe that are using BC30 in their, like I mentioned a few of them, like Unilever, Med Jones, Nutritions, and many of them. I'll just quickly say where we work to produce that. We make sure that it's authentic taste. We make sure that there is less of food waste, and proactive health. We talked about proactive health. There's a lot of research, understanding what consumer need, what customer need, and there's a lot of research that been happening with evolving diseases and the health challenges that we develop in this world when we live here with a lot of stress. At home, at work, how to put our minds at ease, will stay healthy. So there's a lot of work that's been done. And then there is also most of you may be hearing about plant-based. So there's a lot of research that happening with plant-based. So we're also looking at the nutrition of the plant-based, right? So we have many technologies and processes in our sites. As I mentioned, is around 150 sites across globe, and many of them also capable of producing proactive health proteins. So whatever we do is how we produce. There is a guiding principle, the way we produce in our factories or the work we do in research and development. So safety first, quality always. And we always make sure this is a mandate and this is company-wide commitment to ensure that safety of our people who produce our product and also safety of consumer and customer. And there's a different ways we do it. We make sure that everyone is involved in making Kerry successful in terms of safety of people, quality of a product, and make sure that the consumer get what they want. And you see that how we developing those culture across and manufacturing standards. We have a separate work stream for building those culture. We have very strong management systems that take care from development, a conceptualization of a product to develop the product, design the product, and then how we manufacture, and then how we make sure that those are benefits are sustained until their shelf life. So food safety we do by design, right? So it's not by chance. As I always say that, and we partner with many global institutes, and they're already doing their own researches. We get help from them, how we test our product, how we assess that they are meeting those requirements. There are many tests that we do, making sure that our product keep on delivering until the shelf life. So I'm going to talk a little bit more about, so that's merely Kerry, but in Kerry also how the nutritional product and manufacture, right? So I quickly talk a little bit about regulation, which you may be knowing more than me about that. And then manufacturing requirements. We actually do a quite good job in Kerry to ensuring that our manufacturing processes meet those requirements. And we also stay top of the regulations, how we manufacture a product and release our product. And then how we, consumer support in nutritional product that happens. So if you, I know that the team who's listening is actually into this dietary supplementing. And you understand that 21 CFR is DSHIA is the Health and Education Act that actually very prevalent in US majorly. But I think there is various standard around the globe, right? But I think when it comes to comparing and benchmarking, this is mature, but I think there are countries who really done a good job by understanding the basis of what's required. This is a hybrid of between farm and food industry, which I say already. So I think this comes somewhere in between. But I think there are different regulation locally. But I'm only giving reference to FDA, but I think there's compliance to local authorities, like the Canadian authorities measure this under compliance under pharma. China has a different team who look at supplement valuation differently. There are local, like copy of FDA's, they have local regulations. So it's different industry, in different countries around the globe has different standards, but I think they are at par almost with each other. But there are a few differences, definitely. And if you look at this is something I just want to highlight, how you do a dietary supplement claim validation. And again, it's vary from regulation, like claims and dietary supplement in US and Canada is done based on the literature, right, and scientific studies. There are countries, we know that China, Australia, New Zealand, even India, to submit your clinical studies, right? And you may be hearing a lot when we had those vaccines, when we have, COVID vaccines, there was a lot of debate on how the cleaning studies being done, which is more effective. And there's a timeline, what number, how long it takes. There are ways to do it. So there's many standards which talk differently. But I think the global reference always go back to most of the time FDA. But there are local regulation, how you do clinic study and verifications. So Then how you manufacture those, right? This is something important to understand. Joshi, just let me know if I'm overrunning, right? I'll make sure that I'll stay within the time. But if I overrun, just let me know.
Okay, sir.
So the manufacturing specifically, every lot is to be identified and authenticated. And I'll talk a little bit about others in the next slide. What is identification, authentication? But I'm sure that's just nothing not new to you, but I think it's good to have a look at it again for the benefit of those who has any confusion, and we can have a question and answer at the end if you have any questions that you want to talk about. So the ingredient is so important, right? And then we make sure that they're identified, the ingredient, what we want to use, they're authenticated, the purity and the assay that's authenticated. It will be done different ways. I think the technology is evolving. So earlier, I remember my days, we're doing titration, then we moved to HPLCs, GCMS, but there are many ways that different countries at different maturity level do it differently. But there is always way to do it. And then the stability studies for each packaging this is important because we want to make sure that what is the intended use, what we're going to deliver from the product, the day it's manufactured to the day of the shelf life of that product. It should meet those minimum requirements of doses or supporting the clinic study from a day to end. And the way you do it is, again, you maintain those potency of the drug that have been used and in the supplement that get that minimum potency. If you see the labels, it's always say that, what is the nutrition level it should deliver until the shelf life. So it's depending on many factors, how you make your ingredients stable. Sometime you have to capsulate them. You have to make sure that how you do the packaging, what kind of packaging you need to sustain your product to end of the shelf life. And definitely then the next step come when you have all the design part and the ingredients done, then it's a finished product. So every finished product, how you release is actually you need to have there's a protocol. There are in dietary nutrition, there are components sometimes goes as high as 25, 30, and you have to make sure that you have the validation protocols in place. And we'll talk a little bit about that. This is very important. And then going forward, you can reduce your testing by rotational testing that we call, right? And then you decide at what frequency what is to be tested, but it's to be validated first 3 lot where you test everything when you scale up. Then I think there's an important piece for consumer complaints, specifically for FDA. If there's a consumer complaint, adverse reaction, every complaint is to be looked at. And as per FDA, it is reported in back to 10 business days. Right. But this is specifically for USA. There's different companies do it. Countries have different ways of dealing it. But I think the most important piece when you do all of it, how you make sure that when you scale up in quantities like in tons in a factory, doing something in the blending in the lab is different than doing in a factory, which is a huge blender. Sometimes it go as big as 2 tons in carry. And how you're making sure that it will be hygienically produced. So there's a lot of things that come in play in the zoning. So if you don't know, there are zoning of the site, the manufacturing location when you do it. So there's a different way to do it, right? But some of them do it 4 zones, 3 zones. But I think most important is a zone where you can do activities which is not controlled for contamination, like warehouse where the product is not open. And then there's a zone where you do some product opening, packaging, and then there's a zone when you have open product. So all those different zones have different requirements of environment, how you do cleaning and sanitation, what quality of air should get into that, how people has to move in and out from those areas, right? So when you see that, it's few pictures that the team who's manufacturing those kind of product, they're all covered, right? So when they covered, have a mask, no hair, no body part exposed, that's normally to a zone, high care zone or critical zone. Some will say high care, critical, but it can be defined based on your management system. And then what air quality you need, right? Because you have to circulate air, you get the air from outside. So you need to have right level of filters. It could be range from E10, which is, and then go to the HEPA filter, which is S10, right? But there's the pre-filter requirements. So that's a big subject, which need more time to actually go technically deep into this. And how you do cleaning and sanitation. So you don't have cross-contamination. There are companies who just expert in this. You maybe heard of Diversity Ecolab. That's the work they do, right? They know they have different chemicals, how to effectively and efficiently do cleaning and sanitation. Environment program, monitoring program. So you have to keep monitoring your environment based on what the risk of your product. So if you want to make sure that you have environment monitoring plan for pathogens, right? Or you have to look at the microload in that area. Or you need to you need to make sure that what is a pre-operational condition you want to have, what is the operational condition you have. So there should be a complete environment monitoring program to make sure that your process and production environment stay healthy all the time, hygienically, clear, good for the requirement that you have. So you monitor during the process, right? A different way to monitor it. And then you run a production, how long you can run one batch of production. You want to run one batch, clean it, or you want to run 4. So there is a requirement from production run validation. So how long you can run to keep your environment hygienically healthy, right? So that decision will be made based on the environment monitoring. And then you want to make sure that formulation assurance. So whatever you're adding in a product, they should be identified, as we said, right? They would authenticate it, and then the quantity that goes in there, right? You need to have very accurate. So how Kerry does is we have SAP system. So that comes from, small dispensary, small nutrition, built with the bigger, the base ingredients, and our SAP system capture everything that how much you're adding based on the formulation, the recipe that they have. Right. So this is, again, very important thing. If you do everything and don't produce it right, don't produce it, pack it right, you're going to spoil the product. You're going to make consumer sick. And this whole process is not going to help. So this is a very important piece. I talk about this. don't want to take much time, but identification can be done with NIR, FTIR. There's a visual identification. Sometimes there is the organoleptic identification, which we call non-quantitative. And then there's authentication, that how pure it is, right? It could be GC, GCMS. These are being used now by the industry. UPHLC, ultra high pressure, and then HPLCs, LCMS. So there's a different ways you can do it, right? And then there's a test method already there. If you have very complex formulation, then it's a different way to evaluate how you do the finished good testing. Then you have to validate a test method, which is FMOR or ISO equivalent, right? Then you end up doing some equivalent studies for your test methods that meet your local requirement and FDA requirements. So there's this whole lot of stuff that you do for raw material finished good testing. Already talking in the previous slides, but I think this is the main points here is, so one, supplement is qualified. That's, we talk about the validation, 3 successful production run, and then we have then light testing may be optimized once the product is validated. So that means a stabilize, you show that you did all the component testing, evaluated that what we are doing in manufacturing and mixing is actually validated. that means is such our recipe, our formulation, or process of capable of producing every time what is expected. That's the validation, right? And then verification plan you should have by, it could be rotational, right? So in the first validation, when you test all the components, but in the verification process, you might choose on rotation, you want to do the nutrition, or you want to do the vitamins, depending on what you have, or minerals, what you have in your recipes, right? So that's a different way of making sure that your product meeting all the requirement all the time, what is producing on the label or what you're claiming on the label, right? And packaging stability, I think we talked about this one. It's also something very important for each packaging is to be evaluated, because packaging is one of the most important factor to ensure that your shelf life, at the end of the shelf life, your product will deliver what is claimed on the label. Right? So that's important. So what you produce today, sometimes you should see that shelf life 18 months. And there is a study behind that when it's 18 months. That means that what's written on the label on the 18th month, even after deterioration of few minerals, vitamins, it should give you the minimum that's claimed on the label, right? So different ways. Sometimes industry do overdose, but not the overdose that it can be toxic to the consumer. But then there is a way how you do it, right? So there is scientific way to do it. And ICH guidelines also to define about, what 4 different zones. Is based on the climate of the world, right? It's a tropical, what in the center of the earth is a different temperature, different climate, and that have a different ways of doing those studies based on the relative humidity. So different, because at 4 different zones across the world have different humidity, temperature. So ICH guidelines also talk about 4 zones, which is further divided depending on where you're going to send your product.
Sorry to interrupt you, sir.
Yeah.
We are running out of time. Can we conclude it fast?
Okay, I will do it in this. So consumer feedback, adverse reaction screening. So whenever we have consumer feedback, we have to make sure that this is to be all adverse, all consumer complaints should be evaluated and look at what the adverse effect of or adverse reaction. This is a good feedback from the market that we have to keep improving our product. And sometime the regulation want us to report it back, and many a time regulation also has authority to withdraw this product, which has adverse consumer effect. And that's it from my side. Thank you so much. I don't know if you have a time for questions or not, but that's done from my side.
If any of the participants are having any questions, kindly drop the questions in the chat box. So we'll be answering those. Is that okay with you, sir?
Yep. Thank you.
Thank you, sir. Thank you so much for the informative session. Next,
My pleasure.
thank you, sir. Next, I would like to welcome our keynote speaker, Mr. Jason Freeman, founder and CEO, Farmer Direct Organic, Canada. So, over to you, please.
Yeah, thanks for having me. My name is Jason Freeman. I'm founder and CEO of a group called Farmer Direct Organic Foods. And I'm calling you from British Columbia, Canada. I'm going to share my screen now for the presentation, so please bear with me as I figure this out. So, let's see. Is the screen sharing at all?
Yeah, sorry, it's visible.
So you can see, okay, so great. As you can see our logo right here, Farmer Direct Organic. We were In May of 2021, we were purchased by Above Food, which is a large legume commodity trading company in Western Canada. I'm going to be talking about dietary lifestyles and what the organic consumer is demanding in the North American market. So just a little bit about us. We've been working with some of the farmers that we are sourcing from for over 2 decades. What consumers are really looking for now is companies that are working directly with organic family farms, because they're starting to put together that management of the soil is extremely important, or is paramount to the quality of the food they eat. So our brand pillars is we're 100% organic. Again, with dietary lifestyles, these type of consumers are looking for companies that are making 100% commitment to organic agriculture. So organic is all we do. And at the foundation of organics is soil management and building soil organic matter through a number of practices, such as cover cropping, rotational grazing of livestock, and related management practices. This year this January, we became the first organization in the world to launch Regen Organic certified gluten-free packaged oats for the retail shelf. And this is going really, really well. What regenerative organic farming is, it is a certification which takes your baseline National Organic Program. So that's the federal law for organics in the United States as the baseline, then adding certain management requirements, such as testing your soil to see if you're growing your soil organic under increased use of cover crops, increased use of rotational grazing, specific rules in regards to animal husbandry and which breeds and species you're allowed to use under this regime. What they're trying to stay away from is the breeds of livestock that were bred specifically for gains, which many people feel are inhumane. And that's the stereotypical image of a chicken in a coop with their breasts so big that they can't walk. So those types of breeds and that type of animal husbandry is not allowed under organic or regenerative organic farming. We're also certified to Test and Clean, which is a certification regime, and it tests for 200 different toxic pesticides and specifically glyphosate. As in the news, there's been many lawsuits where people have won money against Monsanto and now Bayer Crop Science for contamination of glyphosate, which the courts believe has led to cancer. So again, with the healthy lifestyles, consumers in North America are starting to understand that the health of their food comes down to how it's growing, with a particular focus on soil management. And then the other side is what's not in your food. So we test our food for various pesticides, and everything's family farmed. And in that regards, all of our products are traceable back to the family farm. There's a specific lot code found on our products. And what we're really doing is trying to prevent organic fraud in our supply chains. And we do that through the pesticide testing, the traceability back to the family farm, and the various third-party certifications such as regenerative organic and NOP organic. So again, our standards: regenerative organic certified organic. We're also a non-GMO project verified, and we're also certified gluten-free by the gluten-free certification labeling organization. We're certified tested clean to glyphosate. We're kosher. We're also certified plastic neutral through Repurpose Global. And this is a really interesting company because, you know, our consumers are interested in healthy, you know, nutrient dense food. But they're also very interested in ethics, other ethical considerations, and are looking for companies that acknowledge some of the pollution issues with consumer packaged goods, and are providing solutions for that. So until we go to compostable packaging, and even after that, we'll probably support this organization. For every pound of plastic we produce, Repurpose Global takes a pound of plastic out of the ecosystem, out of the environment from one of their projects in India, and then repurposes the plastic to be used in you know, a product that will last a long time, if not forever. So, for example, furniture made out of plastic composites. And our facility is, and our company is ISO 2200 food safety certified. So that's a HACCP-based system through Brooks. And so, you know, this kind of represents the cutting edge of the market, where you have regen organic certification, traceability back to the family farm, pesticide testing, gluten-free testing, plastic neutrality, and all of these attributes and certifications are really helping propel the brand into the natural products channel in the US. And the natural products channel is comprised of between 25 and 35,000 stores. So individual retail outlets, some are larger chains, many are independent groceries, and this is what they're starting to demand. So again, this is just a comparison between Farmer Direct Organic products and some of the legacy brands out there. As you see, to gain market share, the marketplace is you know, requesting more and more certifications and ethics, especially for new companies that don't have legacy demand. Just one thing I'd like to talk about, Repurpose Global Plastic Neutrality certification. It's much more than a plastic neutrality certification. It's also a poverty reduction and elimination strategy and project. Many of the people they hire are from the Dalit community in India, and previous to working with Repurpose Global, they literally made their living by scavenging for garbage on the side of the road. What Repurpose Global has given them is jobs, salary, personal protective equipment, so gloves, smocks, hair nets, proper bathroom facilities, water, breaks, and all the things that people should have in dignified jobs. So some of the employees of Repurpose Global have reported back, and for one example, one of the women now can afford to pay for her children's education. And as we know, that education is one of the best ways to for people to climb out of poverty and break that intergenerational poverty treadmill. So we're really proud of that certification and being part of that. And again, that ties into dietary lifestyles and consumers that are concerned about nutrition, density, concerned about pesticides. They're also concerned about ethics, plastic pollution, and all these other issues are tied together. So this is just, for example, small innovation our company did. First to market, we just basically combined 3 different types of lentils, red lentils, French green lentils, and black lentils together. And it's an incredibly popular SKU. I'm also going to talk a little bit about organics and organic management practices in a second, but I just wanted to show you a little bit about our brand and how we're innovating. This is just, so this is what our brokers use to sell through the promotions they use. You don't have to pay too much attention to that. but it has a good picture of our products on the retail shelf. For people that are doing product marketing to this type of consumer or any consumer, I really recommend, as you can see with our brand, the logo is very distinct. You can see it from a far away. So I really recommend when you're doing that, make the logo very, very noticeable. The consumer has to see it from 10 feet away, and you should get good exposure. As I mentioned, we innovated this year by being the first organization in the world to launch regenerative organic gluten-free packaged oats, and it's going extremely well. Again, consumers are looking for certified organic brands that they can trust, that they know that it's grown by family farms using sustainable soil management practices. And I just want to go into a little bit about that and why that is so important to the nutritional density of food. So, and why organic farming is the best way to build soil organic matter, build that microbial life in the soil so that you have that synergistic relationship between the plant roots and the soil microorganisms, which leads to nutrient density. So what happens, so through the process of photosynthesis, plants will produce sugars in their leaves. They'll take these sugars and transport them down to the root system. Once the sugars reach the root system, then there's a synergistic symbiotic relationship between the soil microbes, which exchange vitamins and minerals that are in the soil with the plant for the sugars that the plants are producing. When you use synthetic fertilizers, for example, that interrupts that relationship. Synthetic fertilizers, nitrogen fertilizers, interrupt that symbiotic relationship. And so that's why through conventional agriculture, and the USDA has shown this through nutrient density testing since 1950s, common fruits and vegetables are really starting to lose their nutrient density and are about half as dense, nutrient dense as they were in the 1950s. And that's a direct cause of that is through improper soil management, through the use of too many synthetic fertilizers and pesticides. So again, dietary lifestyles, organic consumers in North America are really starting to seek out organic food, regenerative organic food, because they're starting to understand the difference in soil management practices between organic and conventional farming and how that relates to nutrient density. So organic food costs more, but there's more nutrients in it. So if we start looking at the cost versus the amount of nutrients, then it ends up organic food not costing that much or not costing more than conventional food if you look at it from cents per percentage of nutrient density, for example. So here are the 4 regenerative organic certified oat SKUs that we just brought to market. Kind of gives you an understanding of the look and feel of our branding, what's working in the marketplace, and just the look and feel and the aesthetic of our brand. So I'd also like to talk a little bit about our product line and legumes and whole grains. We sell a lot of legumes, a lot of lentils, peas, and beans. These are becoming more popular in North America. Basically, they're fairly new to North America. We don't have a tradition, a culinary tradition, which uses these products. There is somewhat one from the Mexican community who use a lot of beans. But in coming into the mainstream, these lentils, beans, peas are really coming into their own. And so, of course, there's a number of reasons for that. And the 3 biggest reasons are iron, protein, and fiber. Legumes especially lentils, are an amazing source of iron, plant-based protein, and fiber. So of course, that's becoming very, very popular. And also oats have excellent fiber profile. So again, it really fits into dietary lifestyles. So our brand is really catering to that marketplace, catering to those type of consumers. And we're really catering to people who are looking for whole foods. There's a lot of exotic products in the marketplace. For example, probiotics is a good example. And which is great and everything. But for people that are looking for foundational foods, things that you can eat every day that are going to improve your gut biome significantly, whole grains and legumes, lentils will absolutely do that. And this is one of the reasons why they're becoming a lot more popular. So that's my presentation. I think we're pretty, I don't want to cut into the other person's presentation. If there's any questions, I'm more than happy to answer them. If they're not, thank you for inviting me. I've enjoyed some of the talks thus far. Thank you very much.
Thank you, sir. Thank you so much for your informative session. I guess everyone are very keen by listening all the talks.
Yeah, you're welcome. Thank you for having me.
Thank you, sir.
Have a good rest of your conference, everyone. Talk to you later.
Thank you, sir. So if any of the participants are having any queries, kindly drop your queries in the chat box. We will start our technical session by 10:30. As per the agenda, who are the participants who are in the hall A, kindly go for the breakout room and join hall A. And the rest of the participants kindly stay in the main hall. I repeat myself, all the participants who are assigned to hall A as per the agenda, please join the breakout room, hall A. And the rest of the participants kindly stay in the main hall. Before we move to our technical sessions, I request every participant to turn on their cameras for gallery image. I repeat, all the participants kindly turn on their cameras for the gallery image. I request all the lovely participants to have a smile on their face for the beautiful image. Thank you all. If any of the participants facing issue regarding joining the breakout room, kindly let me know. The respective participants of main hall are Bharti Samir Shah, Priya Rani, Aishwarya R, Nasiruddin Khan, Professor Dr. Dilip, Ayana Das, Sweeti Bardhan, Priyadarshini, Dr. Vinita Singh, Garima, Pallavi Ghatia, Dr. S. Shaktiya, Mrs. Sonali Tripathi, Mohan Chandra K. The respective participants of Hall A are Usha Ravindra, Geeta M, Renu, Anushka Khan, Dr. Siddhartha, Sithi Nabla Karim, Soma Srivatsava, Dr. L. Anthony Catherine, Jura Taufka, Sristi, Shreyanshi Singh, Jeena Taman. All the participants I request to join their respective halls. Archana ma'am, can we start with our session? Is it okay with you?
Yeah, we can start with the session.
Now,
Excuse
We
me,
welcome.
am I audible?
Yes,
Madam, please let me know how can I join the hall A?
Okay, may I know which device you are using, ma'am?
I am using a HP laptop.
Okay. As you could see at your top corners, breakout rooms will be available. Click on the breakout room and you could see Hall A. Kindly join the breakout room.
In the top corner, I am having a grid view of participants.
Okay. Give me a moment, ma'am. I will be assigning to you the breakout room so that you could join directly.
Thank you.
Archana, ma'am, you were saying something?
yeah, I welcome everybody in the main hall to present their researches, and we will be enjoying the recent researches in the field of clinical nutrition and also dietary lifestyle. So we will start the session. Just a request that every e-presentation is oral presentation is for 10 minutes, and if there is a poster presentation, then it is for 5 minutes. Kindly stick to the time and we can start the session right from now. The first participant I will be inviting is Ms. Bharti Shah, who will be presenting her topic. Thank you.
Yes, ma'am. Good morning.
Good morning.
Yes.
Your voice is breaking, madam.
Tapi budak tu peramah gila. Semua orang belanja dia. Kita cakap, bukannya saya tak bagi.
Others kindly keep themselves mute, except for Ms. Bharti Shah.
Just a minute, ma'am.
Madam, I have a doubt. How to join the hall A? I am in the main hall.
Yeah,
Ju-
I
just
guess
give us.
there
I
are no
myself Geeta
breakout
Inkanji. How can I join to main hall?
Just give us a moment, ma'am. We will be assigning to you the holiday so that you could join the
As
holiday.
mine is, I think, second presentation.
We have assigned, ma'am. You will receive a pop-up. Kindly check it.
Pop up, madam. Okay.
Good morning, ma'am. Is my screen visible, ma'am?
The screen is visible. Kindly make it to a
Am I audible,
presentation
ma'am?
mode. Yes, you are audible.
So, may I start, ma'am?
Yeah, make it to a presentation. Yeah, perfect. You can
Okay, yeah.
start.
Yeah. Good morning, everyone. Myself, Bharti Shah, my guide, Dr. Jagmeet Madan and Dr. Meeta Singh. So, title of my topic is Assessment of Sun Exposure in Perimenopausal and Postmenopausal Women of Mumbai, Hyderabad, Bidar. It is population-based study. Menopause is considered as a transition phase from reproductive to non-reproductive phase in woman's life. It is characterized as a period that occurs after 12 months of amenorrhea. And postmenopausal osteoporosis also seen in this postmenopausal woman, and which leads to the low bone mineral density and osteoporosis. As we know that osteoporosis is multi-factor condition, but modifiable factors here are the dietary calcium, low dietary calcium intake, and vitamin D, lifestyle factors such as the physical activity, and as well as the non-food source of vitamin D, which is through the synthesis of skin. So now, as per the Indian Menopause Society Guidelines 2020, to get sufficient vitamin D through sun, exposure of 20% of face, neck, both forearms without sunscreen for at least 30 minutes between 10 a.m. to 3 p.m. is required. We know that vitamin D is also referred as a vitamin, but it behaves like a hormone because its production is primarily based on the exposure to sunlight rather than just being obtained through dietary sources. So in India, this while low vitamin D through low sun exposure mainly due to indoor lifestyle habit, traditional clothing, wearing saris, salwars in women, and then darkly pigmented skin and atmospheric pollution. So after reviewing this literature, so need for the study is this study basically undertaken to assess sun exposure in peri and postmenopausal women in different 3 states of India, that is Mumbai, Hyderabad, Bidar. As we know, many hospital-based and clinical studies examine the prevalence of osteoporosis along with vitamin D deficiency due to low sun exposure in general population of India. But very few epidemiological or population-based studies to date done among perimenopausal and postmenopausal women in India. So identifying these contributing factors, osteoporosis and low bone mineral density, which leads to the increased risk factor, especially in postmenopausal women. The targeted intervention is also required for this particular postmenopausal and perimenopausal women, so that they can also get the community-based intervention. So next, moving to my hypothesis, aims and objectives. So is the peri as well as postmenopausal woman getting enough vitamin D, especially through sun exposure, along with the other nutrition factors like calcium intake and protein intake? And so this hypothesis, aim and objective of the study, this study was designed to assess sun exposure in peri and postmenopausal women aged between 40 to 61 years from different socioeconomic strata, that is upper, middle and lower, in Mumbai, Hyderabad, Bidar, by using a validated sunlight exposure questionnaire, which this questionnaire was adapted from Patwardhan and Khadilkar 2018 article. So moving ahead to methodology, this study used the multi-stage stratified sampling design, where total 1,210 peri and postmenopausal women were recruited from Mumbai, Hyderabad and Bidar. This study was approved by the TH Ethics Committee from Hyderabad. My methodology includes basically 3 stages. So total 620 perimenopausal and total 590 postmenopausal women were recruited. In stage 1, a simple random sampling, 5 wards from Mumbai, 2 mandals from Hyderabad, 2 CD blocks from Bidar were considered. Stage 2, again simple random sampling. So a selection of areas were considered. And last stage, stage 3, cluster sampling, selection of location, premises and buildings were considered in Mumbai, Hyderabad and Bidar. So now data collection. So in detail, aims, objectives were explained to participants before data collection. Consent was taken from these participants in English, Hindi, Marathi, Telugu, Kannada language, Telugu in Hyderabad and Kannada in Bidar language. So following such tools were used, and to validate tools, pilot study was also conducted. First tool, basic questionnaire, included demographic information, modified Kupuswamy. Second important tool was the sunlight exposure questionnaire. So validated sunlight exposure based on the Patwardhan Khadilkar article was used, where participants were asked about their sun exposure, their nature of work, job, and sun exposure, especially in minutes per day, and timing were 7 a.m. to 11 a.m. That is one slot. Second, 11 a.m. to 3 p.m. and 3 p.m. to 5 p.m. separately. Additional questions like working, non-working, field work were also included in this study. This is very important part, that solar radiation between 7 to 11 a.m., 3 to 7 p.m. is 40% of radiation between 11 to 3 p.m. So therefore, whatever estimated sunlight exposure duration between 7 to 11 a.m. and 3 to p.m. from the questionnaire was gathered, it was converted to 40% and then added to a sun exposure of 11 to 3 p.m. Now, estimation of skin area percentage based on the Lund and Broader chart, where 5% full sleeves plus cap, 10% half sleeves plus cap, and 15% half sleeves plus no cap were considered for the data collection. My third tool was the OSTA, that is the osteoporosis self-assessment tool for Asians. In this, the calculated by subtracting age from weight and multiplying by 0.2. Then women were classified in the 3 group: high, moderate, and low. And then prevalence of osteoporosis was on this 3 category was considered. Now, last my tool was the SSFQ. From this, mean dietary calcium intake was obtained. Now, postmenopausal inclusion criteria: women who have attained menopause, which is more than 12 months, 40 to 61 years age category. Exclusion criteria: premature ovarian failure, postmenopausal bleeding, or use of any hormonal drug or hysterectomy. For the perimenopausal woman, inclusion criteria: regular or irregular menses, but it should not within the past 3 months. Age category 40 to 61. Exclusion criteria, again, premature ovarian failure or taking any hormonal drugs. But here addition and psychological disorder like depression, anxiety also include. So these were the inclusion and exclusion criteria. Now, result and discussion.
Oh,
So on the basis of result, the average age at menopause and postmenopausal woman was 46.1 in Mumbai, 45.2 in Hyderabad, and 44.9 in Bidar. And majority of the participants from Hyderabad and Bidar, they were non-vegetarian, and only 1/3 of the participants from Mumbai were vegetarian. And Mumbai and Hyderabad women were homemaker, and Bidar participants mainly they were working. Now, this is very important table which depicts the exposure to sunlight in Mumbai, Hyderabad, Bidar. So on basis of this, majority of perimenopausal women, that is 84% from Mumbai, 61.2% from Hyderabad, and 98.5% from Bidar in perimenopause, and postmenopausal 88% from Mumbai, 64.7% from Hyderabad, 98% from Bidar. They had sun exposure less than 15 minutes per day, and therefore they are at high risk. Significant association was also observed between peri and postmenopausal women in Mumbai and Hyderabad, except Bidar. Similar kind of a study was observed by Joeta et al., but they have correlated sun exposure and serum vitamin D level, which was seen very low significant association was seen. This is important result. Now, next discussion, if we can see, India is a very rich sun-rich country, but then also deficiency of vitamin D observed at all age group. Vitamin D is important from dietary calcium, and so for the strong bone health. So this condition entirely preventable by measures like good exposure to sunlight, adequate calcium intake, especially knowing time of the day of exposure to sun and duration, which is very important for sufficient vitamin D synthesis. So my third and fourth research tool. So OSTAS, on the basis of OSTA score, it was found that 22% to 26% of postmenopausal women in these three cities, they were at moderate risk to osteoporosis. It means that as the age advances from peri to post, they are at the higher risk of osteoporosis. From the SSFQ, mean dietary calcium intake was grossly deficient in both groups. So in perimenopausal, just 70% of ER they are meeting, and in postmenopausal, just 50% of ER they are meeting. So why I'm correlating? Because I would like to correlate with the sunlight exposure. So first, I have correlated with the OSTA score and sunlight. So I have seen statistically significant result when OSTA score was correlated with sunlight, and between peri and postmenopausal women, it was postmenopausal women who had less sun exposure, that is less than 15 minutes, less than 15% of exposure of skin area, and hence, so they are at more risk of osteoporosis. So now moving ahead.
Madam, your time is getting up.
Ma'am, this is the last slide, second last slide.
Okay.
So dietary calcium intake is that sunlight exposure is low in postmenopausal women, and dietary calcium intake is also low, so that they postmenopausal women again are low risk. So this is the another study correlated. Now, summary and conclusion, basically more education awareness about the non-nutrition source of vitamin D in peri and postmenopausal women is must, and it is required so that they can get the targeted intervention benefit. My appreciation, I have used Patwardhan and Khadilkar research article validated questionnaire. So I'm thankful to that because it is based on Indian population. Benefits, lacuna of epidemiological data is there. So this kind of a study is required, especially considering three strata. Strength, I have correlated OSTA score and calcium, and my recommendation across pan India along with the therapeutic measures, these studies can be further done. These are the references, and thank you very much.
Yeah, a very good presentation, Ms. Bharti Shah. My only recommendation to you is you should be preparing a nutrition education plan if you have not prepared.
Yes, ma'am. I can't hear you properly. Education plan, ma'am?
Yeah, nutrition education plan, because you have mentioned that there is a need of nutrition education. But if you can attach a nutrition education plan along with this, it will have more credits.
Yes, ma'am. Ma'am, can I say one line? Actually, so I have personally approached to Hyderabad and Bidar and Mumbai, and I have administered this questionnaire, and I have approached the school and colleges where I have seen that many women participant teachers and even in slum area, they were not getting sun exposure because they were not aware about all these things. So I have recommended principal that during call they have an open ground. So I have recommended principal during lunchtime they can ask teachers to just sit, have lunch for 10-15 minutes, not more than that, so they can at least get the this sun exposure. So they have started implementing. They have started implementing by giving notice, and this is the thing the verbally I have explained. But education, that plan what you are asking, that is the my future thing.
Yes, thank you. Yeah. Yeah, thank you, Bharti. Next one is Miss Priya Rani.
Okay, thank you, ma'am.
Ma'am, is screen is visible?
Yes, it is visible.
Okay. Good morning, everyone. My name is Priya Chaudhary. I am PhD student from Punjab Agriculture University, Ludhiana. And today I am going to present my research paper, and the title is Superior Nutrient Dense Germinated Quinoa Variety Formulation of Nutrient-Rich Chickpea for Nourishing Future of Celiac Children. So first I am starting with brief introduction part. We know that quinoa high in nutrient content, especially richest source of vitamin, minerals and amino acids. And most important thing is it is the gluten-free pseudo cereal. So due to this property, it is the boon for the people that are suffering from celiac disease. And we also know that germination significantly improve the nutrient content of the seeds by desirable modification of the availability of the nutrients, and also it can be helps to improve the textural as well as sensory property of the food products. So many studies have been conducted by the plant breeders and showing that quinoa poses vast morphological variation, and germination significantly impact on the starch, protein and minerals bioavailability, and also decrease the anti-nutritional factor that are present in the quinoa seeds. So, what was the need of this study? No doubt, many studies on the quinoa have been done, but nutritional enhancement of germinated quinoa variety have not been studied, and also nutritious germinated quinoa-based product with improved physiochemical as well as functional property have not been developed so far. So, keeping these in mind, the objective of my study to select the highly nutritional dense variety of germinated quinoa on the basis of nutrient quality score. Basically, nutrient quality score is based on the percent adequacy of desirable nutrients. And in this study, I have to calculate the percent adequacy of 11 desirable nutrients and one anti-nutritional factor. Next is to evaluate the sensory property, best cooking method, as well as shelf life of highly acceptable product, and to evaluate the effectiveness of germinated quinoa-based gluten-free chickpea in terms to improvement of health status of celiac children. Now I am coming on the materials and methods. So first of all, I have procured the sample of 10 quinoa variety from the Department of Plant Breeding and Genetics from the Punjab Agriculture University, Ludhiana. After that, processing have been done and processed sample was stored in the airtight container for further nutritional analysis. After that, on the basis of nutritional analysis as well as calculating the nutrient quality score, I have selected the three highly acceptable variety, or we can say nutrient dense variety of germinated quinoa for further product development, and or we can say chickpea preparation. Then for chickpea preparation, firstly roasting have been done of pre-tempered seeds of germinated quinoa with three different methods of the roasting, that is salt roasting, dry roasting and microwave roasting, and making this two different ratio, that is 50-50 and 60-40. After prepare the chickpea and to evaluating the effectiveness of this gluten-free chickpea, I have selected the 90 celiac children in the age group of 7 to 12 years from the local hospitals of Ludhiana, and 30 was treated as a control group and 60 was treated as a experimental group. And supplementation was done during the periods of 3 months along with nutritional counseling, and data regarding anthropometrical and biochemical was collected before and after intervention for evaluating the efficiency of this supplementation. Now I am coming on the results of my study. So as already told that for choosing the highly acceptable variety of germinated quinoa, first I have to calculate the nutrient quality score of all 10 quinoa genotypes and two different age groups. And on the basis of nutrient quality score, results showing that these are three highly acceptable variety of two different age groups. After selecting these highly acceptable variety, these variety further used for product development, or we can say chickpea preparations. And on the basis of organolactic score, it was found that R3 treatment, in which 60% jaggery and 40% dry roasted quinoa seeds treatment highly acceptable. Then this highly acceptable treatment further stored in the HDPE bag during the periods of 2 months and resulted, it was no change in the peroxide value as well as free fatty acid value of this highly acceptable product till 30 days. Then this highly acceptable chicky further supplemented to the respondent, and the supplementation results showing that greater decline in the number of severely underweight subject in the experimental group, and also greater effects on the Z score of the BMI of the age of the experimental group after intervention. And if I am talking about on the of biochemical parameters, so it was seen that significantly improved in the hemoglobin level as well as significantly decrease in anti-TTG level of experimental group after intervention. So it was due to therapeutic effect of a gluten-free, this germinated quinoa-based gluten-free chicky along with nutritional counseling. So now I am conclude my study and and showing that this study helps to identifying the best combination for preparation the gluten-free chicky with best cooking methods and best variety of the quinoa. And this was a perfect chicky for further supplemented to the respondent and showing the best results in terms of improvement of anthropometrical as well as biochemical parameter of the respondent. Additionally, this study also recommendation for the plant breeders and guiding them to cultivation of highly nutrient dense variety of the germinated quinoa on the large scales. And especially as a nutritionist, I have suggested that these are three highly acceptable, or we can say nutrient dense variety of germinated quinoa, so that it should be used for development of nutritious gluten free product with improved physiochemical as well as functional property of gluten-free product. So therefore, it can be helps to tackling the problem regarding low nutritional profiling that are widely low nutritional profiling of gluten-free product that are widely available in the markets. So this is the acknowledgement. Thank you, ma'am.
Thank you, Priya Rani. My only concern is,
Yes, ma'am.
since you have mentioned it can be given as a supplement for
Yes.
low BMI children, but have you calculated the cost effectiveness of that? That is something which is to be done.
Okay. Okay.
Yeah. Because India being a poor country,
Ji.
and maybe through CSR projects or anything like NGOs or anything, you could be doing that, but then you should be having available funds, as quinoa is yet not cheap in the market. So this cost effectiveness has to be worked out very well. Thank you.
Thank you so much.
Yeah, next one is Aishwarya.
Yes, ma'am. I'll share my screen.
Yes.
Is it visible, ma'am?
Yes, it is visible.
Oh, okay.
You have to make it in a share.
Okay. Is it okay, ma'am?
Yes.
Okay.
It is not yet become a slide sharing mode, so you can try that.
Is it not coming, ma'am?
No, no, it's coming as a PPT, but not in a slide share mode. Maybe at the down bottom of your.
Okay, ma'am. One second.
Yeah.
Is it coming, ma'am?
Yeah. Now you can make it slideshow.
Is it okay, ma'am?
It's not coming, but you can start.
Okay, okay. Thank you. Thank you. Okay. A very good morning to one and all present here. I'm Aishwarya, research scholar in food and nutrition. I'm from Government College for Women, Trivandrum, Kerala. I'm presenting a study on the topic titled "Effect of Nutrition Education on Young Sportsperson in the Dietary District, Kerala." So my guide is Dr. Vinny Joseph. She's an assistant professor at Women's College, Trivandrum. So coming to the topic, these are the table of contents I'll be speaking on. So the background of the study, that is introduction. So nutrition knowledge plays a crucial role in the overall performance and the well-being of the sports person. The relationship between the nutrition and the athletic performance is widely recognized, and the understanding the importance of the proper nutrition is essential for the individual engaged in the sports. So the objective of my study, that is relevance of the study, is that furthermore, sports nutrition awareness, attitude, practices among the sports person in the state of Kerala are still unknown. And this study is the first of its kind that examines the nutrition knowledge, attitude, and practices among the sports person in Kerala for a period of 3 months. Objectives. So to assess the socio-demographic and sports profile of the young sports person, and to assess the nutrition knowledge, attitude, and practice of the young sports person. So methodology taken, the sample size was 100, the male was 71 and the female was 29, and the age group was selected from 13 to 24 years, and the one-to-one interview using the questionnaire method was done, that is socio-demographic, sports profile, nutrition knowledge, attitude, and practices were taken. And statistical analysis, mean standard deviation, and paired sample details was taken. So result in the first category, that is social demographic profile, that is 63% of the sports person died in the age group of 16 to 20 years, and 54% of the sports person had a degree qualification, and 69% of the sports person had a nuclear type of family, and 59% of the sports person are living in the rural areas, and the 60% of the sports person had the economic status, that is, for my easiness, I took the ration card color, that is pink color. 60% had the pink color, which states that they were in the BPL, that is priority category. So next category, that is sports profile. Football was the major game played by the sports person, that is 39%, and 33% of the sports person had state level participation, and 65% of the sports person never attended any sports nutrition class before, and 83% of the sports person haven't followed any special kind of diet, and 4% of the sports person, that is 44, that is a mistake, 44% of the sports person had 6 to 8 hours of sleep. And coming to the main part, that is knowledge. So here, the several questions were actually developed in relation to the basic nutrition, proper hydration, and sports nutrition, etc. And these questions were given to the subject expert as well as the non-subject experts. And the question with the highest score was selected, and they were put into the final form. And the final form was then again translated to Malayalam, and it was a close-ended question, that is yes or no, and Likert scale type of questions. And it was an assumption that the higher the scores, the better the participants' knowledge, and attitude, and practice habits will be. So they were also asked regarding the nutrition class. Earlier, I have mentioned that 65% never attended any nutrition class before. So coming to the nutrition part, before the session, the nutrition pre-test was given. Then the nutrition session was conducted for the sports person. And I think they at least took about 5 to 10 minutes to complete the questionnaire. And then the data was collected again immediately after the session, and again after 1 month, and again after 3 months. So these were the procedure maintained. So coming to the knowledge part, here packed sample details was done, and here you can see that there was statistically significant between pre-test and immediately after the session. Then they were not statistically significant, and from the graph it's clear that before the pre-test, that is before the nutrition class, then their knowledge was 69.3, and immediately after the session was 75.2. Then after 1 month, again I have conducted the same questionnaire test with the same sports person. There was a slight decline in their nutrition knowledge, that was about 73.5. Then again after 3 months, again it was understood that again it was decreased, that is 72.3, which actually revealed that the level of retention seemed to have significantly decreased with the time. So that was the nutrition part, knowledge part. So again, it was differentiated between male and female, in which states the male had a good score when compared with the female. And coming to the attitude session, so all the attitude sessions were statistically significant, and here you can see from the graph, it was seen like before the pre-test, it was 78.48, and after 1 month it was like 83.86, which was like big, which proves that the bespoke sessions provided were found to be useful, and they were able to increase the attitude level further. But again, you can see after 3 months, it has again reduced to 80.60, which means that the level of retention is like significantly reduced within the time, and despite the effectiveness of the session provided. So again, gender-wise distribution was done, and it was shown that the male had a good score. So again, coming to the practice session, and here you can see like it was statistically significant, and in the pair 2 and 3, it was not statistically significant. From the graph, it was understood that immediately after the session, that is, it was about 63.8. Again, after 1 month, it was increased, that is 66.26. And again, after 3 months, it was reduced. So here my discussion part is that even though the bespoke sessions proved the effectiveness of the session, they were able to improve their knowledge, they were known to improve their attitude, they were known to improve the practice, but again, with the time, their level of retention seems to have decreased. So my intention is that these sessions and these kind of awareness should be given once in a while. Because to retain this, their knowledge, these sessions should be given in once in a while. And my conclusion part is that by assessing the knowledge, attitude, and practice of the sports person, nutrition education programs and intervention can be developed that addresses the nutrition knowledge, nutrition needs of the sports person. A combination of the solid nutrition knowledge, positive attitude towards sport, and effective dietary practices is essential for the sports person to maximize their performance and health. These are my references, ma'am.
Yeah, thank you so much.
Thank you.
Yeah, Aishwarya, thank you for your presentation. Your study is really good. And also you have concluded that a lot of nutrition education and knowledge has to be given to the sports person.
Yes.
And it is quite eye-opener that people who are in the sports department are not yet providing this kind of a knowledge. And you found around 44% who had no knowledge about it. So that is something quite, one needs to improve on that. Thank you.
Thank you.
Next one, Nasruddin Khan.
We are not having the person. We will move to our next presentation, ma'am.
Okay. Mr. Dilip, Dr. Dilip Goswami.
Yes, madam. Yes, madam. I am here.
Yeah. Thank you so much. You can start presenting.
Okay. Is my slide visible, madam? Hello?
Yeah, now, yes, it is visible.
It is
Put
visible,
it in
yes.
a presentation mode. Yeah.
Yes. Okay. Okay, it is visible now? Okay.
Not in a presentation mode.
Yeah, okay. Okay.
Maybe you can click the F5 to make it the slideshow. Yeah.
Is it okay, madam, now?
Yes. You can start. Yes. Thank you.
Good morning. I am Professor Dilip Kumar Goswami from Sri Sri College of Ayurvedic Science and Research Hospital, Sri Sri University, Cuttack, Odisha. My topic is looking back to explore the concept of balanced diet. This is nothing but some informations that are available in the Ayurvedic classics. And my aim is to percolate the knowledge of the ancient science, Ayurveda, with the modern scientific platform about the concept of balanced diet that can be closely correlated. So the introduction is balanced diet is a concept of food and dietetics that guides about the need of the basic ingredients of emergency sources, the nutrients, body regenerating components that should be present in the food are carbohydrate, protein, fat, vitamins, minerals, and water. Concept of the modern balanced diet, a reverse look on the Ayurvedic classics to understand the concept of our ancestors on daily nutrient or ingredients to maintain health is made in this paper. This is my effort. The present study is considered to have need to understand the knowledge of our ancestors on the composition of diet for maintenance of health. The study is conducted considering the hypothesis as our ancestors were aware about the components of diet and their need for health. The aims and objectives of the study is to explore the ancient knowledge of our ancestors about the concept of balanced diet as mentioned in the Ayurvedic classics. The study was conducted in the Central Library of College of the Sri Sri College of Ayurvedic Science and Research Hospital, Sri Sri University, Cuttack, Odisha, India, by taking the three basic Ayurvedic classics as source: Saraka Samhita, Sushruta Samhita, and Ashtanga Sangraha or Ashtanga Hridaya as the source. Help of internet sources was also taken to some extent to get some idea on balanced diet. The result of my study was, though practically 4 books were studied as the source, then also it was seen that the closely correlated topic on the balanced diet is observed in the Saraka Samhita, Sutra Sthana, that is the Jajjah Purushiyam Adhyayam, slok number 40, as mentioned in the portion of the sloka is Sarva Rasabhyaso Balakaranam. It means taking up all 6 tastes, that is the rasa, as said in the classics, in appropriate manner and proportion among all the ways to get the bala. Ayurveda considers the human body as the combination of 5 elements: Ksiti, the earth, solid components; of water, the liquid components; Teja, fire, the component having the capacity to produce and balancing the temperature; Marut, air, the component that regulates and produces the different movements in the body; and Vyom, Akasha, the places in the body, the spaces in the body that provide lodging of anything. The 6 rasa mentioned in the classics are Madhura, sweet; Amla, sour; Labana, salt; Kotu, pungent; Tikta, bitter; and Kasaya, astringent. Among the 6 tastes mentioned, Madhura, Amla, and Labana give nutrition to Kapha, Siti, and Up, the solid and the liquid portions. Amla, Labana, Kotu gives nutrition to Pitta, that is the Teja, the regulation and production of the temperature in the body. Kotu, Tikta, and Kasaya give nutrition to Vata, Marut, and Bhum, so that regulates the movement of the body. Among the 3 dosha, Vata, Pitta, and Kapha, Kapha is also described as Bala. Bala means strength, immunity, resistance. Hence, need of Kapha in the body is more in proportion. Hence, body needs Madhura rasa in highest proportion. The other 5 rasa is minimal. The need of the other 5 rasa is minimal, but should not be avoided completely. As summary, the concept of the need of all the components available in naturally available foods are the need of the body to maintain the equilibrium of its components. All these 5 tests should be taken, but the proportion should be maintained. Avoiding any of these components is nothing but invitation of the imbalance in the dosage, that is said as dose of Vaishamma, resulting in diseases of different category. In taking the 6 rasa test, the proportion of them should be properly understood and maintained. As conclusion, I want to say a detailed, systematic, and scientific study of the Ayurvedic classics in relation to the balanced diet may be proved to be a boon to the human society. Let it be explored properly. So I acknowledge to our respected Dean Sir for his inspiration to do the academic works and also the International Conference on Clinical Nutrition and Dietary Lifestyle for giving me the chance to present the paper in brief about the concept of the Ayurvedic classics that can be correlated with the balanced diet. And with this, thanks for patience hearing. So this is nothing but an information and sharing of the knowledge. So with this, I conclude because time is very limited, and
Yes.
I hope no
Thank you,
much
sir.
time should be taken. Yes. Okay,
Thank you,
madam. Thank you.
Dr.
Thank
Dilip.
you.
Yeah. Thank you for your presentation. And yes, there is a lot of need nowadays, especially after COVID, that Ayurveda and nutrition should be working together, because nutritional sciences does explain balanced diet very well. And also it is followed by every nutritionist while planning the diet. But when it comes to Ayurveda, there is no binding between nutrition and Ayurvedic knowledge. Maybe in Ahar Samhita dated long back, there is little bit of mention of that, but still it has to be done very properly. So thank you for your presentation. It was a very good topic you initiated. Thanks a lot. And my next presenter would be Ayana Das.
Dr. Archana, just one question.
Yes.
I am Dr. Nasir. Am I in the right hall, main hall?
Yeah, you are in the right hall.
Okay,
so you missed your presentation, but
yes.
Yeah, I guess you can, if you are ready right now, you can start then.
I am ready now.
Yeah, you can start then.
Thank you very much. Just a moment, okay? Can you see my presentation?
Yes, just make it in the presentation mode.
Is it okay now?
Yeah, it is okay. You can start.
Okay. Good morning, everyone. This work is about adiposity indicators and their role in predicting metabolic syndrome in the Arab adults. Actually, in Middle East countries, as you know that there are a lot of problems with obesity and overweight, and that's why the prevalence of metabolic syndrome is actually
category or as continuous variable, make minutes
Hello,
represent.
hello, can you mute yourself other than Mr. Nasiruddin?
Can I continue?
Yes, you can continue.
So as I was saying that the prevalence is really alarming in obesity and overweight among the general population of the Saudi Arabia. Recent studies in Saudi Arabia has shown that there's a high prevalence of metabolic syndrome around 39.8 and 30.4.4 in males. and 29.2% in females, which is really alarming and increasing. This metabolic syndrome is also being reported among adults with type 2 diabetes as well. Anyways, our main aim is here. We are looking for some screening methods which are really easy. It means they can be inexpensive. Okay, they are easy, and they can screen the people who are at risk much before then we find out. So it has been demonstrated that adiposity measurements may prove helpful in assessing and predicting the risk of developing MET-S. For example, there are studies by Amato et al. And Khan et al. In which the VAI and LAP are strong and useful indicators. I will just talk about what are those indicators which I have just put here. Okay. Our aims and objectives. Sorry.
Hello, can you mute yourself? Those who are present in the classroom.
Okay. So till now, our aim and objective of this study are, since there are very few studies in Saudi Arabia comprising many adiposity indicators. For example, here we have included the LAP, which is lipid accumulation product, VAI, which is visceral adiposity index, DAI, ABSI. Similarly, you see at least 7 adiposity indicators has been included in this study, and that makes it a bit unique here, which has not been done in Saudi Arabian population. So our objective was to evaluate the gender-based association and effectiveness of different indices in predicting the metabolic syndrome. And since, as you see in the last paragraph, that the present study choose low-cost, easy to calculate, non-invasive and accessible anthropometric indicators to predict the metabolic syndrome. So the ease of early identification and intervention for at-risk individuals may help control the burden over the country's economic problem as well, and will help to control the severity of the metabolic syndrome. Coming to the materials and method, we have taken 833 participants, in which 10% were male, 15% were female. Age was 42.2. Among them, some of 408 were MET-S and 408 without metabolic syndrome. And these are all taken from our one of the program, which is the Chair for Biomarkers of Chronic Diseases. The participants were asked to fill out a questionnaire which contains information on the demographic and medical history. Non-Saudis were excluded. It means only the residents were included in this one. Our metabolic syndrome diagnosis was based on NCEP criteria, which was based on the elevated waist circumference, elevated blood pressure, elevated FBGs, elevated triglyceride, and low HDLC. Coming to the results, you can see from here, the first one shows that the diagram shows that the chart shows that there's a high prevalence of metabolic syndrome among the male and female here, but you can see that the females are more prevalent here with the categories or with the components of the metabolic syndrome. For example, you can see the reduced HDL or elevated triglyceride, okay, or you can see the central obesity. So it was found that the prevalence was high compared on the basis of the metabolic syndrome. If we are coming to the table, which are the clinical characteristics, then you see that I'm just concentrating on the adiposity indicators which are on the down, which is VAI, DAI, WTI, BAI, ABSI, and BRI. All these adiposity indicators were high in male as well as in female as compared to the control. Among the males and the females, these were high. All the adiposity indicators were very high. Okay? Moving forward, when we have gone to the ROC curve of different anthropometric indicators to check the strength or the prediction of metabolic syndrome of each indicator, we find that the LAP and we find that the VAI and LAP and the BRI were the most one, okay, which were showing the highest one. Okay. the first one is about the overall. The second one is about the male, which shows that in this one also it shows that the LAP and the if we go for the colors, which is not so much clear here, the DAI and the VAI, they were the highest, which have got the prediction for the metabolic syndrome. The similar goes for the female. So these diagrams show that which indicators are more having the more predicting ability to show the risk for metabolic syndrome. We have gone more. We have done more experimental experiments to explain these things. For example, when we have taken or we want to know the association of adiposity indices as predictor of METES, we have done the odds ratio test. Okay, and this also shows that, for example, you see here the LAP in the overall and the WTI in the overall, they were the highest predictor for the metabolic syndrome in overall population. As if we are concerned with the males, then we have the DAI and the VI here. Sorry, here. Okay. Similarly, we can find the female also. Coming to our conclusion, first I will just want to summarize this, that we are talking about here the metabolic syndrome criteria based on the ATP, 833 participant, and we have gone for the determination of the several adiposity indicator, and it has been found that LAP and WTI shows the highest odd ratio. If we go for the discriminatory ability of all these adiposity indicators, and if you want to put them in the descending order, we can see that LAP and WTI were on the highest region. In conclusion, I can say that the prevalence of METES is really high in females than in males, and the anthropometric indicators, which is LAP and WTI, were the better than the other indicators in both the genders, and it is followed by DAI and the VI. Coming to the other conclusion, that the indicators LAP and WTI were better than other in predicting the METES among males than females. So based on gender, the LAP and WTI were better indicator among males as compared to the females. Whereas if we talk about the ABSI and BAI, they were shown, or they were having a weak performance, or they exhibited weak association as determinant for the metabolic syndrome risk among all participants. My last slides will just show the acknowledgement, because I want to thank all the nurses and study coordinators who helped me screen participants. Thank you for your attention.
Thank you, Mr. Nasiruddin. This was a very good study, and more such studies are required in the Gulf region, so that the adiposity can be reduced and metabolic syndrome can be reduced, because that is a major concern in these countries currently. Thank you.
Of course, of course. Any questions, ma'am?
Not really. It's a well written study and well studied program. Thank you.
Thank you. Thank you very much.
So I request Ananya Das to start presenting. Is she present?
Yes, mommy, she is present.
Yeah, okay. Hello, hello. Is there any problem, Ayana? We cannot hear you at all. Joshia, madam?
Yes, ma'am.
Yeah, can you look into this? Because she has
Okay,
not
ma'am.
yet started. Yeah.
Miss Ayana, if you're having any technical issue, kindly let us know in the chat box or on the WhatsApp so that we could help you with that. Ma'am, shall we go for the next presentation until the
She
technical?
has to, she also has to stop presenting then, you know, because, yeah. So next one is Sweetie, madam. Sweetie Bardhan.
Yes, ma'am, I'm present.
Yeah, you can start presenting. Yes, I can see your presentation. You just have to put it in the presentation. Yeah. Yes. Thank you so much. You can start.
Good morning. Myself, Sweetie Bardhan. I'm from West Bengal State University, a research scholar of Food and Department of Food and Nutrition. So, the topic of my today's presentation is comparison between two methods of physical activity assessment in Bengali young adult females and its effect on indicators of T2DM. So we know that the prevalence of overweight, obesity, and other non-communicable diseases are increasing in an alarming rate all over the world, and India is not an exception. And while there is some non-modifiable risk factor of these non-communicable diseases, there are some modifiable risk factors, that is physical inactivity and unhealthy dietary pattern, which contribute a major role in developing these non-communicable diseases. Physical inactivity is the fourth leading causes of death, and 3.2 million global deaths are attributed to physical inactivity. So, the emerging health consequences of physical inactivity have highlighted not only the importance of practicing physical activity, but also to assess the physical activity with proper assessment tool. As we know that there are a number of physical activity questionnaire available in literature, to assess the physical activity. Most of them has designed and validated in developed countries, which may not be relevant in Indian context. So in this backdrop, the objective of my study are to compare the level of physical activity assessed by using International Physical Activity Questionnaire, IPAC short form, and physical activity level on Bengali young adult females residing in and around Kolkata, West Bengal, and to find out the relationship between physical activity and the anthropometric indicators of type 2 diabetes mellitus. For this, initially, the requirements of my study were explained to the individuals, and those who were consented to participate and fulfilled for the study criteria were invited for data collection. Individuals with self-reported health disorder, pregnant women, candidates having locomotor disabilities, and those unwilling to give written consent were excluded from this particular study. And finally, 114 consenting volunteers in the age group of 18 to 24 years constituted the study participants set. And the background information, including age, sex, ethnicity, educational attainment, and family history with regard to diabetes, were collected in a pre-designed schedule. Physical activity was assessed by using International Physical Activity Questionnaire short form in terms of met minutes in a week, as well as from values obtained by calculating physical activity level. The cutoff values for BMI greater than equals to 25 kg per meter square, waist circumference greater than equals to 80 centimeter, waist to height ratio greater than equals to 0.50, and body fat percentage greater than equals to 32 were used to define type 2 diabetes mellitus risk. The findings were presented as mean and standard deviation or proportion. Spearman rank order correlation coefficient is applied to assess the strength of correlation between level of physical activity assessed by using IPAC and PAL. Prediction equation was developed using linear regression model and was validated. Physical activity of the participants in terms of PAL and time spent in sitting leisure time physical activity was distributed among various risk predictors of type 2 diabetes mellitus, and the difference in physical activity between risk groups of type 2 diabetes mellitus was assessed, and p-value less than 0.05 was considered as significant. So here we can see that the mean age of my study population was 19.9 years. And most of them, that is 46% of the total participating individuals were in lower middle socioeconomic class, and 35% of the participants reported to have family history with regard to diabetes. And if we see here in the second table, here we have distributed the time spent in various domain of physical activity in a day. And we have noticed that the maximum time spent was reported in sleeping domain, which was followed by study and leisure time sedentary activity. And among all the leisure time sedentary activities, mobile screening was most common, which was around 5 hours in a day. And the least time spent was observed in recreational physical activity. And almost 35% of the participants have reported to practice recreational physical activity for more than 30 minutes for at least 5 days in a week. Here in this table, we can see that the mean physical activity value when measured by IPAC was 1573, and it is 1.45 when it is assessed by PAL. Next, we have identified or measured the strength of relationship between this two method of physical activity assessment, and we have find a significant relationship between these two method, which was significant at 0.05 level. Next, based on this linear relationship, we have developed a prediction model to predict the values of PAL using the value of IPAC. And here we have also found a significant test-retest correlation. We can see here the correlation value is 0.61 and it was significant at 0.05 level. Next, the values of anthropometric indicators of T2DM of the participant has been presented here. So if we see the mean values of body fat percentage and BMI, we can see that it is 30.13 and 23.71, which is coming under the low risk category, as we have earlier discussed the cutoff values. But if we see the distribution of the participants in the risk category, we can see that 42% and 39% of the individuals were coming under risk category for developing type 2 diabetes mellitus in terms of body fat percentage and BMI respectively. And the mean values of waist circumference and waist to height ratio, which is the most important indicators of abdominal obesity, was 84.8 centimeter and 0.86 respectively, which is already coming under the risk category. And more than 60% of the individuals were having high risk of developing type 2 diabetes mellitus in respect of waist circumference and waist to height ratio. Here, the difference in physical activity between the risk group has been presented here. And here we can see that the individuals with greater PAL values had lower body fat percentage, and the difference was statistically significant. However, we have also tested the difference of physical activity in other predictors of T2DM, that is BMI, WC, and WHTR, but we haven't noticed any notable relationship between the PAL values and these indicators. So we have not presented that in this particular presentation. So as we know that time is a major constraint in epidemiological and field study. And we have seen that performing IPAC require maximum 4 to 5 minutes to complete, whereas the questionnaire for PAL needs at least 12 to 15 minutes to complete. And there is lack of evidence regarding the validation of IPAC in Indian population, and use of PAL value to assess physical activity has been validated in Indian population with large sample size. Coming to limitation of my study, the method of physical activity assessment that I have used in the present study is subjective in nature, which is based on self-report of the respondent, hence may be subjected to recall error and over and underestimation of physical activity and sedentary activity respectively. The number of participants included in the present study was less, and the study was a cross-sectional study which cannot infer any causal relationship between physical activity and risk of type 2 diabetes mellitus. So finally, coming to the conclusion, it may be concluded from the present study that the level of physical activity as assessed by two methods of physical activity assessment has significant positive correlation and has high test-retest validity. IPAC can be used interchangeably with PAL as a valid method of physical activity assessment, and body fat percentage had statistically significant relation with physical activity. So these are some of references of my present study, and thank you for your patience listening. Thank you.
Thank you so much, Ms. Vicky. It was a nice study. And as you yourself have mentioned, that there has to be a lot of, the sample size itself is not very big. And
Yes, ma'am.
also, there are few limitations of the study. So there is a lot of scope in improving the study, and one can come to a actual nutrition knowledge information booklet out of this, which you can publish later on. Thank you.
Thank you, ma'am.
Next is Priyadarshini.
Hello,
Is
ma'am.
she present? Yeah, yes. You can start presenting.
Yes,
Miss Sweetie, kindly unshare your screen, please.
ma'am. I can't share screen, ma'am, because someone is on.
Yes, yes. Just give us a
Yeah.
minute, please.
Just give me some time. I cannot find out the option to stop sharing actually.
Maybe it's on the top of your screen. Just check. Stop sharing. It says stop sharing.
If you couldn't find, try to close the current window so that you could stop sharing the screen.
Ma'am, is my screen visible?
Yeah, you just have to go on a presentation mode. Yeah, yes, perfect.
Okay. So I'm here to present on the topic Ulundakali and a review. Basically a Tamil food, traditional food. So when this nutrition conference was announced, I looked into the topic, clinical nutrition and dietary lifestyle. So I thought of bringing a traditional food which was immense nutritional value, Ulundakali, which was taken as a major food source in ancient days. So these traditional food cultures are now being suppressed by fast modern food habits. So let's dive into it. So the main aim is to review and validate the nutritional value of Ulundakali and suggesting essential needs of traditional food at the time of puberty and fertility periods. So the introduction goes like this. Siddha system of medicine is actually a traditional system of medicine native to India. So this system of medicine places special emphasis on diet and food habits and believes that health and nutrition nourishes the body, mind, and soul. So according to the Siddha concepts, food is responsible for different aspects of an individual, including the physical, temperamental, and mental states. So the body absorbs the nutrients as a result of digestion. But Siddha states that food gets converted into saram. Saram means plasma and then followed by successful conversion into senir. Senir means blood. Und means muscle. And then korupu, that is fat. Enbu, bone marrow, and reproductive elements and body fluids. So as per the Siddha literature, health begins with food. Thiruvalluvar also explains, he is one of the Siddhar. He explains in his Kural under the heading of medicine. No medicine is needed for the body if one eats after knowing that all food already eaten has been totally excreted. So this is the verse which Thiruvalluvar has stated, which means மருந்தன் எல்லாம் யாவைக்கு அருந்தியது அற்று போற்றி உண்ணன். So increased health issue. So why is there is a lot of increased health issue? Puberty is a life-changing time for a teenage girl. Unsurprisingly, research indicates that nutrition significantly impacts the most important aspect of it. Nutritional inadequacies are most prevalent in these days. So it influences not only the adolescent health, but also the risk of major chronic diseases in childhood. So let's see into the other problems. Adolescent problems mainly appears with the gynecological problems. So Ulundakali is majorly a nutritional food for women. It not only helps the women but also the girls. So it helps in strengthening the pubic muscles. First, let's dive into the gynecological problems which are now prevalent in reproductive age group of women. More than 4.5 million women ages 16 to 50 reports at least gynecological condition each year. So half of the menstrual women
Her voice has gone.
Yes, ma'am, I guess there is a connectivity issue with her.
Yeah.
Ma'am, can you hear me?
We can hear you,
Hello?
but your presentation is not seen.
1 second, ma'am.
Yeah.
Ma'am, now is it visible?
Yes, yes.
Okay.
Yeah.
Nearly 2 out of 5 women between ages 14 to 15 experience symptoms of premenstrual syndrome, 10% with severe symptoms. Estimation of approximately 10% of women in reproductive age are affected by endometriosis. So these accounts are around 247 million girls globally and 42 million girls and women in India. About 10 to 20% of women have uterine fibroids and around 5,80,000 hysterectomies are done in US per year. Infertility affected 6.1 million women in 1997 and up from 4.6 million in 1988 all over the world. So for this, the traditional food comes into action. The traditional foods are food which are in their original form as they were created, not modernized, processed, or packaged. They have a long history of supporting good health. Foods that is whole, nutrient dense, simple, and basic, like meat, poultry, eggs, whole grains, fish, beans, and legumes, vegetables, fruits, and all these comes into nutrition food. So in rural areas, people eat foods based on traditional essential foods, whereas the urban population incorporates more modern food in diet. In urban areas, a lot of food is consumed outdoors. So usually in metropolitan cities, women women who are working are actually consuming food, which are mostly on the streets, and they consume food in the restaurants. So these causes a lot of gynecological problems. So which is not the case in rural areas. Food sold on the street by vendors is mostly composed of junk food, polycystic ovarian disease, which have sorry. Ma'am. Excuse me, ma'am.
Yes. Something went wrong, I guess.
Yes, ma'am. 1 minute, ma'am. Ma'am, is it now visible?
Yes.
Yeah, ma'am. So polycystic ovarian syndrome, which has become commonest among young girls in India due to this type of poor nutritional diet. So the essential nutrients during pregnancy, micronutrients need for youth are elevated during adolescence to support physical growth and development. Micronutrients involved in the synthesis of lean body mass, bone, and red blood cells are especially important during adolescence. Iron. So iron requirements are increased during adolescence to support the deposition of lean body mass, increase in red blood cell volume, and need to replace iron loss during menses among females. Iron needs are highest during periods of active growth among all teens and especially elevated after onset of menses in adolescent females. Calcium. So calcium needs are higher because of accelerated muscular, skeletal, and endocrine development. Bone mass increases in puberty. Females acquire around 37% of total skeletal mass from ages 11 to 15. Calcium-rich foods include milk, yogurt, cheese, soy products, and kidney beans. So in vegetarian cases, calcium supplement is required. Zinc helps in build-up and repair of DNA. A flourishing immune system ensures that body stays strong enough to grow and develop properly. Zinc is essential to this process. Zinc-rich foods, fortified cereals, and poultry, lean meals, all those. Next comes folic acid. Folic acid is the need for folate increase during later adolescence and to support accretion of lean body mass and to prevent neurotube defects among females of reproductive age. So folate-rich foods includes dark green leafy vegetables and citrus fruits. Then protein. During adolescence, protein requirement vary with degree of physical maturation, and protein needs are determined based on body weight during puberty. These help bones, organ tissue growth. Essential fats includes omega-3 and omega-6 essential fatty acids. So these are vital for hormones, skin, nail and hair health, energy, brain function, and cardiovascular health. So some of these are acquired by oily fish, pumpkin seeds, sunflower seeds, flax seeds, and all those. So what are the effects of processed foods? PCOS in reproductive age group women, uterine fibroids, early menopause, and infertility emergence. So here comes the main food, ulunakali, black gram. So it is one of the healthy dishes given to girls during puberty in villages in earlier days. It is traditionally eaten with jaggery and gingerly oil. It is considered to be a storehouse of proteins. So urad dal actually strengthens the uterus, It reduces the pain during menstruation and uterine wall contracts. So it enhances digestion and also reduces the cholesterol. It helps to strengthen the pelvic and backbone. And usually its main function is to strengthen the pelvic muscles. So the ingredients of ulundakalli includes ulundu. Ulundu is black, urad dal, and gingerly oil, jaggery, and water. So ulundu, according to the Siddha context, botanical name is Vigna mungo, and in vernacular language we call it ulundu, madam, and marsham. So some of its actions include demulcent, refrigerant, aphrodisiac, galactogogue, tonic, and nutritive. So these verses are from Agatya Gunapadam. These verses are taken from Siddha's context. Siddha named Agatya, who mentioned about ulundu. So this verses tells us that it boosts mineral density, bone mineral density, and it's good for skin. It improves digestion and acts as a diuretic. And mainly it strengthens the pelvic floor muscles and boosts energy. Usually, women in earlier days used to consume ulundu at least thrice in a week, but it is not the case right now. Next comes yellennai, that means gingerly oil. So the botanical name, this is extracted from L, that is Sesama indica blend. So some of its actions are demulcent, laxative, nutritive, and emollient. So these verses from Agatya Gunapadam tells us that it acts as demulcent and it is given for cleansing and purgative action, good for digestion. It boosts the heart health. Next comes, so these are nutritional value which are taken from research papers. Hello? Hello?
Yes, you can carry on.
So next, Nalla Vellam, that is jaggery. So these are extracted from Saccharum officinarum, vernacular name Karumbu and Vellam. The actions include demulcent and antiseptic. So these verses tells us that it acquires digestive stimulant that aids in improvement of digestion and appetite, recommended for migraines. It helps in cleansing the blood clot from the body during pregnancy. It regulates blood pressure and heart function. So these are the nutritional values. It includes particular minerals like calcium, magnesium, potassium, phosphorus, sodium, iron, manganese, zinc, copper, chloride. So these are the quantities in milligrams, and some of the vitamins include A, D2, E, and all those, and trace amounts of vitamin B1, B5, and B6. So in larger quantity, we see vitamin E around 113. So jaggery also gathers a considerable amount of ferrous salts during its preparation, as it is prepared in iron vessels. This iron is also good for health, particularly for those who are anemic or lack iron. Jaggery also contains a traces amount of mineral salts which are beneficial for body. Furthermore, jaggery is very good as cleansing agent. It cleans lungs, stomach, intestines, esophagus, and respiratory tracts. So the preparation method of Ulandan Kali is that first the urad dal is roasted for in a low flame for 5 minutes and it is grind well. Then it is in a pan we add palm jaggery and some amount of water to melt it. And if the jaggery is completely dissolved, it needs to be checked for the consistency. And we have to add a cup of water to it. And now the urad dal powder is being added to it. And we should ensure that no lumps are formed, so that the next procedure comes into action is that on a kadai we used to use a wooden ladle. We use tudupu for kali. So now in low flame we have to keep stirring without leaving. It will start thickening by itself, and then slowly we have to add amount of tablespoon of oil, that is gingerly oil, and then we have to prevent it from sticking on the sides. So when it starts to stick, add remaining amount of 1 tablespoon oil, and keep cooking. So then we have to roll like a ball and it can be served. So the nutritional value of Ulandankali, around 100 grams of Ulandankali is approximately equal to 1056 kilocalories. And it is rich in iron, calcium, protein, sodium, potassium as mentioned earlier. So from this, we come into a conclusion that food is an essential source of power. It supplies more nutrients for health. Also, it is a symbol of hospitality and friendship throughout the world. It is an outlet of emotion. Food is a source of security for people to feel reasonably secure when they have enough food stored up to take care of them during periods of scarcity. By taking traditional foods, it helps to regulate menstrual cycle, facilitate normal vaginal delivery, and reduces the risk of early osteoporosis and postmenopausal symptoms. And these are my references.
Yes, thank you, Priyadarshini.
Thank you,
It
ma'am.
was, it was nice to know the benefits of traditional food. But I feel that this study, one should be, substituting palm jaggery to sugarcane jaggery, because palm jaggery is not available everywhere. So if you can try that with substitution and see what if the nutritional content of the product is differing or not, and also the acceptability of the product. So one study can be dedicated to that. And another thing I would like to also add that if this is beneficial along with the modern food, because modern food, as you yourself have said, that in the coming era or in the present era, since women are working, most of the people are consuming ready to eat or maybe processed food. So is there any beneficial effect in spite of eating the processed food? Has to be checked as well.
Ma'am?
Yes.
Ma'am, there's a lot of nutritional
I
value, ma'am.
understand. I understand. But when somebody is consuming traditional food with this, then definitely there are lots of benefits. But those who are consuming, I mean, one has to study that, like how beneficial it would be, even if somebody is consuming processed and packaged food, but also consuming this. So that has to be studied separately, I think.
Yes, ma'am. Yes, ma'am. I'll continue it, and I will precisely do the study, ma'am.
Yeah, maybe there is a further scope for this study. Yeah, next is Vinita.
Thank you, ma'am.
Yes, ma'am.
Vinita?
Yes, ma'am.
Yeah.
Yes, ma'am.
Ma'am, before that, can we ask Ayana Das for the presentation, if it's okay? Okay, I guess
Yeah,
Vinita ma'am has
Vinita
started. We will.
yeah. After Vinita, maybe we will do Ayana Das.
Okay, ma'am. Thank you.
Okay. Dr. Vinita?
Yes, ma'am.
Yeah, you can start.
My screen is visible?
Yes, ma'am.
Okay, ma'am. Good morning, everyone. I am Vinita Singh, working as Assistant Professor at Krishi Vigyan Kendra Gautam Budh Nagar under Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut, Uttar Pradesh, India. So, I am giving oral presentation on the topic ensuring adequate nutrition to prevent childhood malnutrition. So we all know that food security is a vital issue of present time. So first, regarding food, which is provided and supplied through various modes, and its security exists when all people at all times have physical, social, and economic access to food. It must be available in sufficient quantity and quality to meet the dietary needs and food preferences, which is supported by an environment of adequate sanitation, health services, and care, allowing for a healthy and active life. And according to National Family Health Survey 5, which was conducted in year 2019 and 21 in India, nearly 36% of the children below 5 years were stunted, having a short height according to their age, and more than 19% of the children were wasted, having low weight for their height, and more than 32% of the children were under the category of underweight having low weight according to their age. So every man, woman and child has the right to be free from hunger and malnutrition in order to their development and maintain their physical and mental status. But at the present time, more than 800 million men, women and children are denied the most basic human rights of all, that is the right to food. So food alone is not sufficient to secure a sustainable, satisfactory nutritional status, but the different aspects of health must be considered. So food and nutrition security achieved if adequate food as quantity, quality, safety and socio-cultural acceptability are available and accessible and satisfactorily utilized by all individuals at all times to live a healthy and happy life. And the nutrition focus adds the aspects of caring practices and health services and healthy environments. This aims at what is more precisely called nutrition security, which can be defined as adequate nutritional status in terms of protein, energy, vitamins, minerals for all household members at all times, and thus in principle is more than food security. So, ensuring food security and providing nutritional security in a sustainable manner is one of the most towering challenges facing by us today. Food and nutrition security are broadly categorized by three pillars as food availability, food accessibility, and food utilization, that is food absorption as nutritional outcome. So first, which is availability of food, that include the production of food having different determinants as the easy availability of food to every household. Second pillar is easy access to food, which may be defined in terms of purchasing power of the households. And the third one is food utilization, or we can say as food absorption, which implies that the person being able to assimilate the food eaten to lead a healthy and long life. So there are various factors which affect the food and nutrition security. Some of them are like poverty, poor market accessibility, lack of proper knowledge, increased price of essential food items, etc. So basically, a healthy Indian diet focuses on cereal grains, pulses, vegetables, fruits, healthy fats, dairy and spices. So we need a wide range of nutrients to lead a healthy and active life, and for providing these nutrients, good nutrition or proper intake of food in relation to the body's dietary needs is required. So an adequate and well-balanced diet combined with a regular physical activity is a foundation stone of good health, and the poor nutrition can lead to reduced immunity, increased susceptibility to chronic diseases, impaired physical and mental development, and reduced productivity. So a healthy diet consumed throughout the life course helps in preventing malnutrition in all its forms, as well as wide range of non-communicable diseases. But rapid urbanization, increased consumption of processed foods, and changing lifestyles has led to a shift in dietary patterns. So sustainable diet is generally healthful and it has low impact on the environment and food supply. So adopting a sustainable diet will be helpful in maintaining the human health, and it also making sure that the planet has enough resources to feed future generations. And the concept of sustainable diets is not new, but it is complex. And in this simplest term, it looks to have a positive impact on the individual and environment, both now on and in the future. So to promote all the dimensions of individuals' health and well-being with a low environment impact are easily accessible, affordable, safe, equitable, and culturally acceptable. So diet can be made more sustainable by making healthier food choices, by reducing wastage, which can lower the global GHG emissions up to 50%. And the consumption of seasonal low-cost produce like fresh fruits, vegetables, fruits, nuts, and seeds can be increased in our daily diet. So healthy dietary practices should begin early in life, because the undernutrition in many set the speed for diet-related chronic diseases in later life. So the healthy diet consists of different kind of food, and the emphasis has been shifted from nutrient orientation to the food-based approach. So foods can be categorized according to the functions as energy-rich foods, which are carbohydrates, and fats like whole grains, cereals, millets, vegetables, oils, ghee, nuts, oil seeds, sugars, etc. And bodybuilding foods are protein. So the pulses, nuts, and oil seeds, milk and milk products, meat, fish, poultry are very important to include in daily diet as it is source of protein. And the protective foods, which are rich in vitamins and minerals, are required in very small amount for the body, but very essential for performing as a part of various vital functions of our body. So basically, the seasonal fruits, the green leafy vegetables, roots and tubers, and other vegetables are only the source of providing very essential nutrient, vitamin, and mineral. There are several consequences of nutrition insecurity which can be seen throughout the life. This picture shows that the pregnant women with under nutrition give birth to a baby with low birth weight and impaired mental development, characterized as malnourished, started from childhood and continues throughout their life, as they suffer in all the stages like child, adolescent, adult, and elderly with multiple nutritional deficiency disorders and various diseases. So this vicious cycle will again give a birth to a baby of lower birth weight and so on. So these are the different strategies which can be helpful in preventing the malnutrition among children, like well-designed food-based interventions combined with appropriate supplements and supported by health and nutrition education, which can improve the diets of vulnerable population in a relatively short period of time. So, a more sustainable approach for subsistence farming households is diversification of their food production, which can fulfill the macro and micro nutrient deficiencies in the local diet. So, it can be concluded that the nutritional security should be achieved through both the quantity and quality of food products. And important strategies to improve the nutrition security are like promotion of industrial fortified products, promotion of enhancement of diversified agriculture production, empowerment of rural women, providing nutrition education, etc. So, complementary nutrition and therapeutic supplementation for children and fortification and supplementation for mothers should receive priority in this regard. So in order to tackle widespread problems of nutrition, it might be necessary to move in nutrition intervention that addresses the immediate determinants of nutrition and devote new and aggressive focus on nutrition that again addresses the underlying causes of undernutrition. And promoting food security offers various opportunities to increase financial and development in cost efficient ways. Thank you. Thank you.
Archana ma'am. I guess there is a connectivity issue with the ma'am. We will wait for 2 minutes.
I am Garima Bhatnagar. I go next.
Just give us a moment, please.
Okay.
Archana ma'am, are we having you on the line? Ma'am, your voice is not audible actually. Ma'am, your voice is not audible actually. Ma'am, try to rejoin.
Okay.
I have dropped the Facebook and YouTube links. I request all the participants to follow and the subscribe for more updates about the conferences. I repeat myself, I have dropped the Facebook link and the YouTube link of our BioLeaks page for more content and about conferences. Everyone could follow and subscribe those pages. Thank you.
Hello. Hello.
Yes, ma'am, you're audible now.
Yeah. What happened now? Is Anaya coming? Sorry, Ayana Das. Is she coming?
Yes, ma'am, she's coming. No questions for Vinita, ma'am.
No questions for her.
Okay.
Yeah.
Dr. Vinita, kindly unshare your screen, please.
Okay, ma'am. Thank you, ma'am.
Miss Ayana Das, you were saying something?
Yes, ma'am, I'm presenting now.
Okay.
Ma'am, after Anaya, I go next. Garima Bhatnagar.
Yes.
Yes.
Okay, thank you.
Your screen is visible.
Okay. Am I audible? May I start?
Yeah. You have to speak little louder.
Okay. So, good noon, everyone. First of all, this is Ayana Dash from the Department of Food and Nutrition from West Bengal State University. Today, I'm here to present a topic which is entitled as you can see, that a study on the assessment of nutritional status of Bengali children of age 7 to 9 years. As we all know, that the children regarded as the most valuable asset of any nation embody the future citizenry, and they have the crucial potential for the ongoing national development. This phase of childhood is characterized by shift and intensive development influenced by the interplay of genetics, socioeconomic, and environmental factors. Among all these, the nutritional status of a child emerges as a pivotal determinant, significantly impacting their overall health. Optimum growth and development outcomes are always linked with adequate nutritional status, forming the cornerstone for their holistic development. So, as we all know, that the nutritional status or the overall nutrition play a crucial role for the entire or the overall development of a children. So now there is a prevalence of malnutrition. As all we know, that malnutrition is a generic term. It means either it also comprises two different forms. One is undernutrition and another one is overnutrition. And in case of undernutrition, we know it always means there is a lack or deficiency of one or more nutrients, or inadequate absorption, or the presence of different diseases that make it difficult to absorb the adequate amount of nutrients in someone's diet. So, in one hand, malnutrition means that there is a deficiency of nutrients, and on the other hand, it also a determinant that there are some condition where the excess consumption of nutrients is also can be seen. So, this termed as overnutrition. And there is a prevalence of malnutrition in children in the country in the several forms, contrary to the popular belief. In one hand, there is overweight and obese children, and worryingly, the number of such children is on the rise. Accordingly, estimates from the International Association of the Study of Obesity and the International Obesity Task Force, almost 200 million children are now categorized as either overweight or obese. Meanwhile, 47% of the Indian children are undernourished, and which is highest in the world. So, in this backdrop, the main aim of this study is being undertaken to assess the nutritional status of children of 7 to 9 years of age. Now, the methodology. The present study is being conducted on the Bengali children aged between 7 to 9 years in South Bengal district on obtaining the prior informed consent from the authorities of the school that they have attended or their parents after explaining the study objectives and the requirements. The data of 62 Bengali children aged between 7 to 9 years have been presented herein below. Using anthropometric measurement set, data on parameters such as body height, nearest to 0.1 centimeter, and the body weight, again nearest to 0.1 kg with light clothing by using an electronic weighing scale, were obtained as required to fulfill the study objectives. Apart from body height and weight, the waist circumference were also obtained by using non-stretchable measuring tape, and the body heights and weights were standardized into specific categories by using the ICMR reference value, and the BMI for age was analyzed. Waist to height ratio was calculated along with the macronutrient intake of the participants by using the Indian food composition table by NIN. Now the results. Here in this table, we can see there is age in the completed years. I have already told that I have taken the 7, 8, and 9 years of children in my study here. And this age, this child table is mainly represented the BMI for age for my subjects, depending on their categories and their reference value I have used here. It is recommended by the WHO. And here I have shown that how many children are categorized according to the different subcategories of the BMI range. Here we can see that the every BMI range is divided into 5 subcategories, ranging from severe undernutrition to moderate nutrition to normal to overweight and lastly the obesity. And in the age of 7, 8 and 9, every category is divided into these 5 distinct categories with their reference values. Here in the age group 7, I have found that a generous population is found that they are in a normal category, but there is a evidence that a little bit of amount of children there is a risk in developing obesity in the near future. When we're talking about the age group 8, here we can say that majority definitely are under the normal category, but there is a chance that they are also developing the obesity and the overweight category. In the age 9, I can see that majority percentage are normal, but there is an increasing trend to developing obesity in their current years. Now, in this table, we can see that there is a ratio between their waist circumference and height. According to this ratio, whenever a subject's waist circumference by height their height is categorized within 0.50, then it is considered as normal according to their abdominal obesity. But if this range is if this cutoff is reaches to 0.50 or above this, then it is categorized as that person is either have abdominal obesity, or he or she can have a chance to or risk to develop abdominal obesity in his or her recent futures. So here in this category, we can see that at the age of 7 years, near about most of the children are under the normal category. Only 2 subjects are there who are at the risk of developing abdominal obesity. In the age of 8 years, we can see that the only 4 subjects are there who are at the risk of developing abdominal obesity. And surprisingly, in the age of 9 years, we can see near about 6 subjects are there who are at the risk of developing abdominal obesity. So gradually, with increasing age, we can see there is a increasing risk of developing obesity also according to the BMI chart. And here, according to the waist circumference by height ratio, it can be also stated that there is a trend that increasing risk of abdominal obesity is also increasing according to their age factors. Now, here in this table, we can see that this table mainly represented the nutrient content of the subject of the study subject. Age group ranging 7 to 9 years, and here the actual intake of the subject is represented in the terms of mean and SD value, and which is again here standardized by using the RDH chart given on 2020. And here I have taken the 5 categories like the energy, carbohydrate, protein, and fat. In terms of energy, we can see the RDA value is near about 700 kilocalorie, whereas in case of my subject, actual value in case of my subject, it is found that it is near about 1650 kilocalorie. And in case of carbohydrate intake, we can see that 100 gram of carbohydrate consumption is recommended, whereas the mean value shows the consumption is near about 192 gram. In case of protein, the consumption value is 90 gram recommended value, and the actual consumption is a bit higher than the actual value, that is near about 40 gram per day, which I have found in my study. And in case of fat, the recommended value is 30 gram, but eventually it has been seen that in my population subjects, their daily requirement or their daily consumption is near about 20 grams. So from there, from this thing, it can be say that It can be said that anthropometry contributes to a prevalent approach for the evaluating and tracking the nutritional well-being in both individual and community. And it is crucial to actively promote and the sufficient intake of essential nutrients among the children, and emphasizing the role of different primary care and school in the direct influencing their overall growth and development. And beyond nutritional status, the different other factors like the school environment as well as family environment, community settings stand out as a significant role or play a significant role as a educational sphere where the children acquire their healthy eating and lifestyle habits. And here are the different references. Thank you.
Thank you so much, Anaya. It was Ayana, I guess.
Yes,
Ayana?
ma'am. It is
Yeah.
Ayana. Yeah.
Ayana. Yeah. And it was a nice presentation.
Thank you, ma'am.
And although we say that it is a double sword malnutrition in our country,
Yes.
you have studied in children. That is something which is very good. Thank you so much for your presentation.
Thank you, ma'am.
Yeah. Next one, Garima.
Yes, ma'am. I'll just start sharing my slides.
Yeah.
Ma'am, you cannot share. Yeah, okay. Is my slide visible?
Not yet.
Okay. Give me a moment. Are they visible now?
Yes. You can put it in a slide share
yeah,
mode.
yeah.
Yes. You can go ahead.
A very warm welcome to one and all present. I am Garima Bhatnagar, research scholar enrolled in Dalbagh Educational Institute, deemed to be University Dalbagh, Agra, and I will be walking you through my presentation, which is a review about plant-centric diets for India, a trifecta of public health, planetary well-being, and sustainability. Here, I will be sharing some overwhelming scientific evidence which will increase our trust in plant-based diets, and which is important for Indian scenario and how we can achieve it. We know that food is the single most lever to optimize human health and environmental sustainability in Earth. However, humanity is facing a big challenge about healthy diets, providing healthy diets from sustainable food systems to everybody, and food is currently threatening both people and planetary health. We also heard our keynote speaker, Dr. Rao, yesterday, who pointed out that poor diets are the main cause of deaths all over the world, to the level of 11 million people die every year due to dietary risk factors, which we know are modifiable also. And in India, the scenario is not much different. India, being the fastest growing economy of the world, houses 1.3 billion people in the world, but the level of the ranking of India is in global hunger index is 22nd, which is quite high, indicating food insecurity. And India also is burdened with triple malnutrition triology, which is undernutrition. Many children are wasted and stunted, overnutrition, where one person in two is obese in urban India, and 2/3 of Indian population has essential micronutrient deficiencies. And if we take a dig closer, we find that there is a lot of evidence stating that India is consuming foods which are rich in processed meat and ultra processed foods, which are causing these diseases. And the NCDs, which are the metabolic syndromes, are also rising because of the dietary factors leading to cardiovascular disease, diabetes, and hypertension. Thus, India need to have a transformative approach towards sustainable diets. And what is sustainable diet? These have 4 components. They have to be nutritious, affordable, environmental friendly, and culturally acceptable. When we shift to sustainable diets, then only we can achieve the goal of sustainable development goals of SDGs of 2030, where it will provide good health to every individual in India by meeting the RDAs. The diet is culturally acceptable, traditional, seasonal, and locally sourced, and also is affordable, reducing hunger and improving the food security, as well as when it is environmentally sourced and friendly, we will be able to improve our life, the biodiversity of species below water as well as on land, and thus achieve something solid in climate action. As I was mentioning, that plant-based diet, thus the main focus should be plant-based diet in India. If we see, the plethora of studies have mentioned that plant-based diet are rich in micronutrients. They have a lot of antioxidants. They have phytochemicals, which all help us to have a good health, both physical and mental health, to the extent that when depressed postmenopausal women were prescribed healthy plant-based diet, they show better improvement in their psychometric scores, and depression was at bay. Thus, we can say a plant-based diet not only nurtures mental and physical health, but also have 4 other components like a sustainable diet, which are environmental, economic, and social. As I was mentioning that because of the phytochemicals and all the functional food components in the plant-based diet, the diets of plant-based are better in providing alpha and beta diversity of microbiome in person, and thus all these are conferred by reducing, and the good microbiome helps in confers in reducing several risk factors of diet like stroke, ischemic heart disease, total cancers, improves mood, and also it decreases all the biomarkers for inflammation and oxidative stress. It does not stop here. Economic factors also help when the person is happy and healthy. There is less when you are healthy, there is less expenditure in people consuming plant-based food when compared to non-vegetarian food consumers, to the extent that in the world, $216 billion dollars are spent every year if the person is consuming a non-vegetarian diet. But in plant-based diet, there is reduced medical expenditure, medical admissions and x-rays, fewer surgical procedures, medication prescribed, and also there is less wastage when you eat vegetarian foods as compared to when consumed non-vegetarian food, where around 30% of food is gone in wastage. Thus, plant origin foods are less resource intensive, because the emission of greenhouse gases and less footprints of water and land is used, which improves, if you see the social and inner component, food security in people. The amount of food we grow, in India, only 2% of land is available of the world to grow food for agriculture, and we have 1.3 billion population. So if we have to grow food for the non-vegetarian like livestock, then we have to give a large amount of our land. Therefore, if that is converted back in the world, 4 billion more people can be fed if we only grow food for humans. This supports that we develop as a nation animal welfare, support the foundation of peace and compassion, which is an inner component and not looked by the SDGs. And it also ensures family welfare when you have good health. But if you see the Indian scenario, the level of vegetarian has reduced from 25 to 90%, as shown in the 2019 Nutrition and Health and Family Survey, National Family Survey. If we see the analysis of the current diets, we will find that the diets of Indians is lacking in 11 out of 25 nutrients, of which, if you see, these food groups which are protective are consumed below the optimum intake, but the intake of oils and sugars is quite high, and is the ultra processed foods in the society is also high, though the animal foods are low, but they if you see, this is reflected in the slide, which shows that the fiber, vitamin A, riboflavin, and potassium are abysmally low in the diets of Indians because they don't consume fiber-rich food, vegetable and fruits, and nuts and oil seeds. This is reflected in the health of the environment, which says that India, in spite of consuming less animal products, have a very high emission from mutton, which is a meat product, and also from processed food, which are consumed and penetrating every section of the society. Though milk and milk products are also consumed in India, but if you see, there are only 5% of those, GHGs, that is the greenhouse emissions, are only 5% of the one which is produced by 1 kilogram of animal fat. Why is meat so intensive? If we see that animal products are, if I consume 100 calories of wheat, I'll be emitting, there will be an emission of greenhouse gas to the tune of 14 to 12 kgs. And also, I'll be requiring around 1,200 liters of water for beef or pork to produce one serving. But on the other hand, when you take vegetarian diets, there is 30 to 50% reduction in environmental footprints, be it land, water, or energy sources. Thus, adopting vegetarian diets ensure public as well as planetary health. The other component of sustainable diet is affordability. If we say Indians spend around 40% of their food, of their expenditure on food, however, there is, which comes mostly, and the energy comes mostly by the cereal, but they have a reluctance towards vegetable and fruit section, though they can, even in spite of having limited amount, they spend a lot on meat products, both in rural and urban areas. And the amount of junk food consumed is also alarming, as 17% of calories come from junk food, and 30% of fat come from junk food. And if we translate it, we will find that 1.5% more Indians will have to shell out if they want to shift to sustainable diets. Thus, meat eating is becoming difficult and is a hard ingrained habit. There is lack of knowledge about the relationship between food, environment, and health, and thus the reluctance towards learning negative impact of food. And also, there's a faulted belief system that best source of protein is meat, but we don't. And also, we don't prioritize the sustainability, but we prioritize our taste, convenience, and price. Also, meat is subsidized by most of the governments globally after sugar and rice. So how can we counter this? There are several interventions can be seen. As meat is subsidized, we should subsidize plant-based foods and make it more easily available. We should make meat and processed food more taxable, and leaders should champion the cause of plant-based diet and planetary health. Meat and its adverse effects should be highlighted because many are not aware. And also, we should, as a nation, should develop more better tasting meat substitutes which are juicy and match the texture of meat. Last but not the least, behavior change communication strategies which promote dietary diversity will improve micronutrient adequacy and diet quality. Last but not the least, I have to mention that once India transitions to a more plant-based diet, all will not be in the initial years will not be rosy. As when India moves to a vegetarian diet, there will be an increase in greenhouse emissions, water and land use, especially in the population, the poor population which is consuming less than their RDs. But the national averages will substantially vary, will cancel the variation or mask the variation, and will decrease in meat-based and processed food diet. Also, this will help us to achieve 70% of our SDGs by 2030, and India will become a healthier nation with better food security and longevity. Thus, India, with its huge population with rising income, if continues its business as usual model of food consumption, undesirable impact will be felt with both locally and globally. By demonstrating the multifaceted advantages of plant-based diet in addressing India's complex challenges, this paper provides robust scientific evidence for the policymakers, educators, and healthcare professionals to advocate for implementation of sustainable dietary habits towards a healthier and more resilient future of India and the planet. By putting coordinated actions, India can thus achieve its target of United Nations Global Sustainable Goals, and this, backed by vegetarian diet pattern, evidence-based, will help India turn into a healthy, sustainable, and more essentially a consensus society. Thank you for your attention.
Very good. It was a nice presentation and you summed it up also very well. Thank you so much, Garima.
My pleasure, ma'am.
Yeah. Now next participant is Pallavi.
Am I audible, ma'am?
Yeah, you are audible. Your time is 10 minutes. Thank you.
Thank you, ma'am.
Oh, yours is a poster presentation, Pallavi?
Yes, ma'am.
Then 5 minutes.
Okay, ma'am.
Yeah.
Very good afternoon, everyone. Myself, Pallavi Gadia, PhD scholar in Ramadevi Women's University, Bhubaneswar, Odisha. My poster presentation is on nutritional challenges and morbidity status of children in tribal communities, a systematic review. As we know that children are the most vulnerable human resources of our society because they are the future of our nation. But it has been seen that a significant numbers of children are not benefiting equally from the India's tremendous economic success since independence. And various researches shows that they comes from the most deprived segment of our society, which is known as the tribes. And there are over 700 tribes in India, and there are 104 million tribal children in India, from which 20 million are malnourished, which has negative influence on their survival, development, educational performance, and future production. The previous researchers also discovered that a child birth in a tribal household in India had 19% greater chance of dying in neonatal stage. And what are the causes? Because of the negligence, poor nutrition, and different morbidities or diseases. Here we can see what are the nutritional challenges and the morbidities in tribal children. My methodologies are: all the information was gathered from secondary sources only, such as the books, journals, and websites published between the year 2013 to 2023. And all the studies reviewed have been conducted in different areas of India. So here is my results. The table number 1 depicts the nutritional status of tribal children by underweight, stunting, and wasting. So we know underweight is weight for age, and stunting is height for age. and wasting is weight for height. And here we can say different authors or different researchers are compiled in this table. And here we can see the underweight is in percentage. We can see this, it is very much high, which can ranging from 35.9 to the highest is 63%. And we can see in stunting there is, it's ranging from 16.2% to 62%. And wasting is also high in tribal communities, which is ranging from 9.8% to 54%. And what are these causes of this type of malnutrition in tribal communities? These are because of the poor quality of diet. As we know that we need all type of food, all the basic food groups for our proper development of our health. But in tribal communities, they are not aware about this, and they're taking only a few types of only cereals or some types of pulses, millets, and they are not aware about this. That's why they are having this malnutrition. And there is also this cause of the illness or diseases which um, uh, which um, um, which restrict the absorption of the nutrition and cause malnutrition. There is also this is because of the cause of poor maternal health and socioeconomic status. Like tribals are mostly in low and middle socioeconomic status. That is why they are mostly suffering from malnutrition. Here in table number 2, we can see the different morbidities of tribal children. Here I have taken 5 studies where we can say there are different morbidities like anemia, diarrhea, gastrointestinal infections, dental problems, infestation, and respiratory infection. These are the common morbidities in tribal children, and we can see the respiratory infection is highly found in all type of researches. This is because of it can be a cause of poor housing or poor environmental condition in the tribal areas. And here I have figured, I have created the graph of respiratory infection among tribal children. We can see that high amount of respiratory infection can seen in tribal children. So we can conclude it like this. Review sheds light on morbidity pattern and nutritional challenges like malnutrition and in the form of underweight, stunting, wasting among tribal children in India. The key findings are that the tribal children face a range of health challenges and morbidities like anemia, diarrhea, gastrointestinal infections, and respiratory infections. Yes, ma'am.
Nothing. You can continue.
Okay, ma'am. So here are some recommendations like an encouragement should given to the researchers to do continuous researches and monitoring to understand the evaluating or evolving health challenges in tribal communities, or implementing targeted nutritional intervention to address the highest rate of malnutrition among tribal children, and conduct awareness campaign on preventing different morbidities, and emphasize the importance of sanitation, hygiene, and timely medical interventions in tribal areas. And my acknowledgement is this poster was fully advised by my supervisor, Dr. Jyotirmay Udgata. Thank you, ma'am.
Thank you so much. It was a nice poster.
Thank
We
you,
can
ma'am.
move further to Sakthia. Yeah, next candidate. Yeah.
Good morning to all. Ma'am, my voice is audible, ma'am?
Yeah, it's a little louder, madam.
Okay, ma'am.
Yeah.
Good morning to all. Thank you so much for giving this opportunity to present my paper. My topic is myself, Dr. Shaktiya Sivakumar, Professor, Department of Obstetric and Gynecology, Sairam Homeopathic Medical College, Chennai. My topic is nutritional impact of millets, improving iron status and hemoglobin level, body mass index on pregnant and lactation mothers. Pregnancy demand specialized nutritional requirement for mother to maintain good health and during pregnancy and aid in proper health and development of the fetus. Millets are known to help increase blood production, nourish the spleen, and stimulate appetite during pregnancy. Millets are powerhouse of essentials such as fiber, complex carbohydrate, minerals, and vitamins, which is essential for growth and development of the fetus. Millets are rich in iron, calcium, folic acid, and nutrients that are vital during pregnancy. These nutrients help in preventing anemia, ensure the proper bone development, and support the growth and neural tube in the fetus. Nutritional value of millets for the pregnant woman. Every lady requires special nutrition for a special time for ladies. Pregnant women want high fiber. Millets in high in dietary fiber, which help in prevent constipation, common issue during pregnancy. It also promote healthy digestion, help regulate blood sugar levels. Natural source of folate. Millets contain significant amount of folate, also known as folic acid or vitamin B9. Folate is essential for proper development for baby's neural tube and help prevent neural tube defects like spina bifida and any other congenital abnormalities. Millets are also rich source of iron, which is essential for formation of red blood cells in the fetus. Millets also contain vitamin B6, which help in the formation of
क्या हुआ?
Can I continue, ma'am? Formation of neurotransmitter in the brain of fetus, support weights management. Millet having low glycemic index, which means it releases the glucose slowly into the bloodstream, preventing sudden spikes in the blood sugar. This characteristic help in maintaining healthy weight during pregnancy and managing gestational diabetes mellitus. More common problem nowadays, every pregnant women met the gestational diabetes. Antioxidant properties. Millets are rich in antioxidants such as phenolic compounds, which help in reducing stress and inflammation. Antioxidants are beneficial for overall health of the mother and developing the baby. Gluten-free alternative. Millets naturally gluten-free, making it suitable option for pregnant women with gluten intolerance and any other gastric disorder. Nutritional values of millets. Pregnant women, moreover, millets are rich in iron, essential for protection of hemoglobin in the body. Hemoglobin is responsible for carrying oxygen to the cells and body tissues, including growth of the fetus. Iron deficiency anemia during pregnancy can lead to anemia, which causes fatigue and weakness and many other complications. Therefore, including millets in the diet can help pregnant women meet their daily requirement and prevent anemia. These nutrients are essential for healthy growth and development of the fetus, can also help in maintaining overall health of the mother. How millets help in maintaining healthy weight gain during pregnancy? Weight gain during pregnancy is normal and healthy, but excessive weight gain can lead to complication. Millets are low in calories and fat, making them ideal food to add to pregnant diet. They have high satiety value, which means they keep you full for longer and preventing overeating and excessive weight gain. Iron help formation of red blood cells, which calcium, magnesium are essential for development of the strong bones. Role of millets in preventing anemia during pregnancy. Millets are excellent source of iron, which help in preventing anemia during pregnancy. Millets also rich in other essential nutrients such as folate, magnesium, zinc, which are crucial for healthy development of the fetus. Folate help in preventing birth defect, while magnesium aid in formation of bones and teeth. Zinc, on the other hand, play a vital role in immune system, helping the growth of development of brain, baby's brain and nervous system. Material and methods. My material source of data collected from OPD and IPD and Peripheral Health Center of Sri Sairam Homeopathy Medical College and Research Center. Medical camp conducted by the college. The literature will be collected from various authentic medical and diet book and website. Case performer, pre-designed case sheet, nutritional assessment prepared for the topic, diagnostic criteria according to the clinical presentation and lab investigation, physical examination, HP percentage, and BMI. My research design, prospective clinical study design without control group, sample design, purposive sampling done as per inclusion and exclusion criteria. Sample size will be minimum 30 in number. Inclusion criteria, patient of different age group, every 15 days once follow up. All the cases will be followed for the period of 3 to 6 months duration. Exclusion criteria, patient under any other system of medication, people who are having associated with other complaints, especially GERD. Material and methods, primary and secondary data collected through the pre-designated case sheet from 2022 to 2023. Data on weight, BMI, hemoglobin were collected. Measured monthly weight gain through the adult weighing scale and height through the stereometer during the baseline. This measurement was used to calculate the BMI using standard formula of weight in kilograms. Data on hemoglobin were collected by official laboratory technician using Salis hemoglobinometer. Result, that mean weight gain for pregnant women 3.63 plus or minus 5.29 kg with mean hemoglobin improvement 0.5 plus or minus 6 gram per deciliter. Similarly, mean weight gain for nursing women while 3.0 plus or minus 6 kilogram. Mean hemoglobin improvement 0.0 plus or minus 1.6 gram per deciliter. This study indicating positive impact of this millets food on weight and BMI. Discussion. Millets are used to because they have high nutritional properties. They contribute improvement of weight and hemoglobin. As I mentioned, that result same improving the level of hemoglobin and weight in both pregnant and nursing mother. The limitation of this study was sample size due to small sample size due to droplet. This droplet occurred majorly due to socio-cultural reasons such as migration and family tradition. Conclusion of my study, the study showed excellent source of iron with a low cost potential for reducing iron deficiency anemia for pregnant and lactating women. This millet-based food have positive impact on nutritional status of pregnant and lactating mother. In conclusion, incorporating millets into pregnancy diet can provide numerous health benefits for both mother and the fetus. Thank you.
Yeah, thank you so much, Dr. Sakya. My only question is how many times in a day millets were introduced to the pregnant women, or maybe how many times in a week?
Ma'am, this is not measurement, ma'am. Instead of taking our regular diet, we can take the millet-based food. It will be more beneficial for compared to normal diet, ma'am. Instead of taking normal diet, we can take that millet-based food, anything, ragi, anything, whichever available, and will be low cost compared to other non-vegetarian and vegetarian food. Millets will be low cost and highly effective. Nowadays, we are using so many millets, compared to our normal rice, wheat, and all. So, especially pregnant and lactating mother, we can use. It will be helpful for a healthy baby and a healthy mother also.
Okay, thank you so much.
Thank you. Thank you so much.
Yeah, next one, Sonali.
Is my voice audible, ma'am?
Yes, audible.
Thank you, ma'am. And ma'am?
Yeah, you have 10 minutes.
Just network issue, ma'am.
Yeah, so can we start with some? Sonali madam, are you doing it?
Ma'am, just network issue.
Okay, so can we start with somebody else?
Yes, ma'am.
Yeah, so Mohan Chandra.
Yes, ma'am.
Yeah.
Okay, ma'am. Ma'am, is my screen visible?
Yes.
Hello, this is Mohan Chandra from St. Joseph College of Engineering. I'm doing my BTech Biotech. This is my final year project with my co-author Tejashwini. My project topic is development of health drink mix to prevent acidity. Introduction. Acidity has become increasingly prevalent in today's fast-paced lifestyle. Individuals experience discomfort and health challenges associated to it. Nowadays we have our lifestyle has changed very much, that we don't eat proper meals for timing. Like we eat most of the time we are taking dinner after 10, which is very harmful to our health. We should take our So because of this, many young people are facing stomach acidity, which leads to ulcer without treatment leads to stomach problem, and it also can reach to stomach cancer. Neglecting acidity compresses digestive health and may lead to more severe condition. This is an innovative formulation, the power of carefully selected natural ingredient, creating a blend that addresses the acidity and promotes optimal digestive wellness. Our main objective is develop a health drink mix. The drink is assessed aiming to provide a holistic and scientifically supported preventive solution for acidity related issue. Why we did this drink? Most of the antacid present in the market has magnesium compound and aluminum compound, which cause diarrhea and constipation. This also interfere with the absorption of phosphates in the formation of bones. Our project plan, we collected fruits, then literature surveys, we extract by ethanol, then we did antioxidant assays. Materials and methodology. Ethanol extraction, we took fruits, pomegranate, muskmelon, papaya. We choose these three fruits because pomegranate has a characteristic which inhibit ulcer causing bacteria. These three fruits are alkaline in nature. Ethanol extraction we used for 72 hours. Then we did antioxidant assay, which are DBPH, phosphomyelidium assay, Fe2 reduction assay. Phytochemical estimation we did with tannins, phenols, and flavonoids. Antifungal assay and antibacterial assay, we used bacteria which normally spoil the food to check the shelf life and the antibacterial activity of the other. We have done GCMS analysis and we have done nutritional estimation for the product. Result and conclusion may be explained by my co-author Tejashwini.
Good afternoon, everyone. This is Tejashwini, final year biotech student. I'm a co-author of this project, Development of Health Drink Mix to Prevent Acidity. We have performed the assay, antioxidant assay. Oxidation is a process by which a chemical reaction that produce free radicals, which may be a potent carcinogenic. These antioxidant activity help to prevent the oxidation. We have performed the antioxidant assay like DPPH, then phosphomolybdenum, superoxide radical scavenging assay, and Fe3 plus reduction assay. And the results were very promising. The pomegranate shows the maximum oxidation antioxidant property. Then estimation of phenols, tannins, and flavonoids. These phytochemicals are also very essential constituent of diet to prevent acidity. We have also performed assay on inhibition of Helicobacter pylori
which causes ulcer. And then the nutritional estimation of the compounds, pomegranate, papaya, and muskmelon have been shown here. And then the antifungal and antibacterial assay were done with normal food spoilage bacteria like fungi like Candida albicans, Candida krusei, and tropicalis. And then common bacteria which causes spoilage of food like E. coli, Staphylococcus aureus, Pseudomonas, and Bacillus subtilis. These are the results, and pomegranate shows promising inhibiting nature, which can be seen in the antibacterial and antifungal activities. And we have finally formulated our drink by calculating the IC50 values. And the pomegranate and we have mixed the fruits in this ratio of 3:2:1. And we have further given for GCMS analysis and waiting for the results. This is our product formulation, which is basically a syrup. We have sun-dried papaya and muskmelon. Pomegranate was not easy to sun-dry, so we have took a juice of pomegranate and finally formulated into a syrup. Honey is used as a preservative and have shown promising activity to prevent acidity. This is the nutritional estimation of a formulated drink. It shows calories 45 calories, carbohydrates 10 grams, sugar 6 grams, and proteins, fats, and every potassium 160. The conclusion, it is an innovative approach where the fusion of traditional knowledge and modern science and preventive health care. And then we developed this mixture for a flavorful health and promising health benefits. We are further planning to commercialize this product, and we hope to commercialize it. Thank you.
Thank you, both of you. Excellent work you have done in this field. My only question is, what is the shelf life of this?
Actually, we have formulated and it has been 1 month and it's still active. We are further testing it.
And acceptability is good?
Yes.
Okay.
The drink is 1 month and it's fine actually.
Okay. Second is, you said that papaya powder and melon powder, but not pomegranate. So that fruit should be available throughout, right?
Sorry, ma'am, come again.
You mentioned about pomegranate juice.
Yes,
It's
ma'am.
not powder.
Yeah,
So
actually we couldn't sun-dry pomegranate.
you couldn't do it.
Yeah.
Okay, so you still can try on that.
Yes, ma'am. Thank you.
Yeah.
We are looking for lyophilizing technique to try it.
Which technique?
Lyophilize.
Okay. Okay, good work. You have to patentize this.
Yes, ma'am. We are working on it. It's our final year project actually. We are on the progress of work actually.
Yeah, very good. Excellent.
Thank
you so much. Next one is Sonali ready?
Ms. Sonali Tripathi. Are we having you on the line?
Yes, ma'am. Ma'am, due to network issue, my poster is not viewing on screen. Can I just save my reviews?
Yeah, no problem.
Okay, ma'am. Thank you. Okay. Thank you, ma'am. Good afternoon, everybody. This side Sonali Tripathi, PhD scholar of Ramadevi Women's University, Bhubaneswar, Odisha, India. First of all, I thank USF and members for giving me. Okay, ma'am, just screen sharing.
Yeah, we can see your screen.
Okay, ma'am. Thank you.
Yeah.
Just I scan. Okay. Okay, thank you. Good afternoon, everybody. This side Sonali Tripathi, PhD scholar of Ramadevi Women's University, Bhubaneswar, Odisha, India. First of all, I thank USFN members for giving me a chance to present an e-poster. My topic is Health Issues of Transgender, a Comprehensive Review. So as we know, transgender is an umbrella term for all people whose gender identity does not match with the sex assigned to them at birth. The objective of the study is to identify and understand their physical and mental health. All the data are collected in secondary form, such as books, journals, and etc. We can see figure 1. 30% trans feminine are living with HIV, while 70% have high chances of acquiring HIV due to lack of awareness. In figure 2, while comparing health issues of cisgender versus transgender, we can see transgender are more prone to asthma and poor mental and physical health. Then in table 1, the data says that 19 number of self-employed trans have low stress, while 37 have high stress. 18 number begging trans have low level stress, while 38 have high stress. Then 16 sex workers are trans have low stress, while 31 have high stress. Then finally, I conclude that trans community has high risk of HIV and other sexually transmitted infections. So specific program should be implemented by government. Thank you all for patiently listening my presentation.
Thank you, Sonali.
That is my e-poster.
Yeah, it is a e-poster. Yeah, you did well. Only thing is you have not mentioned the, any strategy for the government.
Okay.
Yeah, you have said that government should be implementing certain programs for transgender, but there is no strategy which you have given. That is something which is lacking. But otherwise, it was good. Thank you so much.
Okay, thank you, ma'am.
Yeah, so Joshia, I guess we have finished with everyone.
Yes, ma'am.
Yeah, so I'll be sending the report shortly, but I must congratulate everyone because everyone has actually done very well with innovative projects and new knowledge in the field. Thank you so much.
Thank you, ma'am. Before everyone leaves, I request everyone to turn on the cameras for the gallery image again. Before we conclude the session, I would like to thank each and everyone who have participated actively in this conference, and I thank all the session chairs for giving their valuable time to judge all the presenters. Coming to next session, coming to the next point, we have another conference coming up. Which is 7th International Conference on Food and Nutrition in the month of June. If any of the participants are interested to join the conference, kindly contact Ramya ma'am, or else I will be sharing you the conference link in the chat box. Everyone could join that. Coming to vote of thanks. Thank you is such a prayer that cannot be seen or touched. It must be felt by heart. I feel honored and privileged to get the opportunity to propose a vote of thanks on this special occasion. I would like to thank all the honorable dignitaries, committee members, and invited speakers who blessed us with their presence. And I also thank to all the attendees for both the days of the conference. If time is money, then today you have millions spent million of us. Thank you to all of you for making this event successful today. And for all the participants will receive the certificates in 4 to 5 working days via mail. If anyone is having issue regarding anything, please contact the coordinator. Thank you. Thank you all. Have a good day ahead.
Thank you.
Hello.
Yes.
Madam, I wanted to ask that the judges have graded us on our presentation, right?
Yes.
So how we will come to know about that?
You will receive the winners list while receiving the certificates itself.
Okay, okay, okay. Thank you, ma'am. Thanks a lot.
Thank you. Have a nice day.
Can
Thank
we
you.
leave?
Yes, ma'am, please.
Thank you.
Thank you. Thank you.